Good afternoon, Ohuna. Can you hear me? Am I audible? Ohuna?
You are audible.
Okay, fine. ঠিক আছে। ভিডিওটা একবার দেখে নেওয়া যাক। হ্যাঁ, মাস্কটা খুলে দেবো, বাট ইটস ওকে আই গেস, রাইট?
হ্যাঁ ম্যাম, আপনি অডিবল, আপনি আপনাকে দেখতেও পাচ্ছি, ঠিক আছে।
ওকে।
Am I audible?
Yes, ma'am, you are.
A very good afternoon to one and all. Welcome to the fifth chapters of Frontiers in Biological Sciences, FIBS, organized by the Department of Microbiology, St. Xavier's College, Autonomous, Kolkata. FIBS is an annual seminar of the department which aims at expanding the knowledge of the students regarding recent advances in the field of biological sciences. This year, due to the current COVID-19 scenario, the seminar is being conducted online in the form of a webinar with an international flavor to it. This year, we will have speakers from all around the world, naming University of California, San Diego, University of Chicago, as well as the reputed Scripps Research Institute, USA. And we are proud to announce to you that all of them have been students of St. Xavier's College, Kolkata. Now, I would like to request Professor Dr. Shudeshta Shamchodhuri ma'am, Head of the Department of St. Xavier's College, Autonomous, Kolkata, to invite Reverend Father Dominic Xavier SJ, Principal of St. Xavier's College, Kolkata, Autonomous, to inaugurate the webinar by delivering the welcome address. Over to you, ma'am.
Thank you, Anonna. So good afternoon, everyone. It is my privilege to welcome our Father Principal, Reverend Dr. Dominic Xavier SJ, to inaugurate our webinar, Frontiers in Biological Sciences, Chapter 5, organized by Microbiology Department and supported by DBT Star College Scheme, Government of India. Father, I welcome you to bless us with the inaugural speech.
Thank you very much. Very good afternoon to all of you, to all the respected speakers, professors, and students who have joined us today for this wonderful initiative by the Department of Microbiology, the Frontiers in Biological Sciences. I feel immense pleasure to welcome you all to the fifth chapter of this one-day international webinar. As we have traversed through the new normal, technology has enabled us to always keep on learning and expanding our knowledge base, and this webinar is an example of the same. This platform provides a wonderful opportunity for students to become familiar with advancements in science being carried forward by eminent researchers across the globe. We are honored to have respected keynote speakers who will be enlightening and illuminating as on cellular survival strategies, signaling also liver fibrosis, as we endeavor to better understand the biological and biochemical processes that constitute our being. I am particularly happy to note that all the speakers are our ex-students, and after their PhD, they are all engaged in cutting-edge research in biological sciences in different institutes in the USA. I strongly believe that through these scientific deliberations and interactions, our present students will be immensely benefited. In a very special way, I would like to welcome all the speakers of our students, our old students. We are very, very happy to welcome them, and we are also happy that they are able to give us the time to share their research knowledge to our students. I would like to once again take this wonderful opportunity to welcome all the eminent speakers, members of the staff, participants, and students, and hope to embark on a successful journey where we all widen our scientific horizons. Wish you all the best. May God bless you. Have a wonderful afternoon with our own speakers, our eminent speakers, our old students. Thank you very much.
Thank you, Father. Thank you for your blessings. Without your support, it is not yet possible for us to organize such webinar. Respected Father Principal, respected Vice Principal, Dean of Science, Dean of Arts, our honorable speakers, professors, scholars, support staffs, lab assistants, students, external participants, and my dear friends. I'm glad to take this opportunity to welcome you all to the departmental webinar Frontiers in Biological Sciences, Chapter 5, supported by DBT Star College Scheme. Department of Microbiology is the department focusing on the practical application of theoretical knowledge. Maintaining a good teacher-student bonding, it tries producing scientific papers and products, keeping in mind the broader aspect of life. The professors and scholars being the support system of practical knowledges, the approach to science is transdisciplinary, giving the equal emphasis to biostatistics, biophysics, biochemistry, as well as bioinformatics. We are happy to host our ex-students as our speakers who are involved in cutting-edge research, serving the bigger society in their respective esteemed laboratories abroad. Dr. Shaurodipto Ganguly, postdoctoral scholar of University of California, San Diego, will talk about the understanding of liver fibrosis, a multidimensional integrative approach. Dr. Asif Ali, postdoctoral scholar of University of Chicago, will deliver his talk on the sticking together in subcellular space to survive against races, an inbuilt biomolecular phase separation program as cellular survival strategy. Dr. Shreyushi Chatterjee, postdoctoral scholar from the Scripps Research Institute, USA, will deliver her talk on the biochemical aspects of the physiological condition in specific biological system. Finally, on the behalf of entire teaching fraternity of the department, I would request Father and everyone present to bless all the organizers for the successful journey of this webinar. Welcome you all. Thank you.
Thank you, HOD ma'am, and thank you, Father Principal, for such a wonderful inaugural ceremony. Now, I would like to invite Professor Debjani Dr. ma'am to deliver the vote of thanks and end the inaugural ceremony. Over to you, ma'am.
A very good afternoon to all present here. It is a great honor for me to propose the vote of thanks for this inaugural session of Frontiers in Biological Sciences, Chapter 5, 2022, organized by the Department of Microbiology, St. Xavier's College Autonomous, Kolkata, and supported by the Department of Biotechnology, Government of India, under the DBT Star College scheme. First of all, I would like to give my hearty vote of thanks to our beloved Father Principal, Reverend Dr. Dominic Xavier, SJ. Thank you, Father, for your wonderful inaugural speech, which I'm sure is always motivating and encouraging for our entire department. Thank you, Father, for taking out time for this session so that we are always blessed by you and all the fathers. I would next like to thank the Vice Principal, Arts and Science, Professor Bertram Da Silva, for being with us. I extend my thanks to the Dean of Science, Dr. Tapati Dotto, and my thanks are also due to the Dean of Arts, Dr. Orgho Banerjee, for their constant support. Thanks are also due to the Secretary, Academic Council, Dr. Oyan Chandru, and the IQAC Coordinator, Dr. Parthupatim Ghosh, for always being with us. Sincere thanks to all the speakers who definitely are Xavierians. So we all welcome you and definitely thank you so much for agreeing to share your valuable knowledge with us. I am extremely thankful to Dr. Arup Kumar Mitro, convener of the program, and Dr. Shudeshna Shyam Chaudhary, the head of the department, who have taken all the pains to look into the finer details of the event to make it a great success. I sincerely thank all the professors, support staff, students of the microbiology department, both UG and PG, and the scholars of the department who have played crucial roles in various aspects for organizing this event. It has been a pleasure to host this event, and I sincerely thank each one of the participants. I'm very much thankful to all the sponsors and technical support team, and also to the MC who have responsibly taken up the task beautifully organizing the event. Last but not the least, I thank one and all present, involved, and organizing this session so that it's a grand success. Thank you.
Thank you, ma'am. Thank you, Father. Thank you, HOD, ma'am. Now we would like to start with the first technical session of Frontiers in Biological Sciences, Chapter 5. I would like to call upon our moderator for this session, Professor Dr. Kosturi Sarkar, ma'am, to introduce our first speaker of the day, Dr. Shourodipto Ganguly, a postdoctoral scholar from the University of California, San Diego. He would deliver a presentation on the topic Understanding Liver Fibrosis, a multidimensional integrative approach. Over to you, ma'am. Due to some technical reasons, Dr. Shourodipto Ganguly is not here with us yet, but we are hoping he'll join soon. Please be with us. And I would also like to announce that we will be circulating the feedback form soon. So please keep a check on the comment box. Sorry to keep you waiting, participants. I would like you to request to please like, subscribe, and press on the bell icon for our YouTube channel so that you receive the updates whenever we are online for any seminar from the Department of Microbiology, St. Xavier's College, and you get an opportunity to participate in these amazing seminars and also enlighten your knowledge regarding biological sciences. Our speaker for the day, Dr. Shourodipto Ganguly from the University of California, San Diego, will be here with us soon. He's facing some technical issues. Please stay with us.
Good afternoon, everybody. We are about to begin our technical session, and our first speaker is Dr. Shaurodipto Ganguly, who is currently a postdoctoral scholar at University of California, San Diego. Before moving to University of California, he did his PhD from University of Kolkata, and his area of research was mechanisms of cigarette smoke induced pulmonary vascular fibrosis and emphysematous. I think I'll learn about it from Dr. Ganguly more. Alveolar damage. And currently he is working on understanding the molecular mechanism of liver fibrosis and its progression to hepatocellular carcinoma. Today he will be speaking on understanding liver fibrosis, a multidimensional integrative approach. Dr. Ganguly did his bachelor's in science and master's in science from Bangalore University, India. And before that, he was a student of St. Xavier's College, where he completed his plus 12. So welcome to your alma mater, Dr. Ganguly. He has received several awards, and some of them are really prestigious. One such award is Early Career Investigator Award in Basic Science, American Association for the Study of Liver Diseases Foundation in 2021. Another award I can see here in your CV, Jutica Research Fellowship from Calcutta University. And he has published several good papers in peer-reviewed journals, and I'm happy to yes, the papers are really wonderful, and they have been published in PLOS One journal, PNAS, Analytica, Chemia Acta, Gastroenterology, Nephron, Hepatology, which is a very prestigious journal, also in Cellular and Molecular Gastroenterology and Hepatology. And he has served as an invited presenter in many occasions, in many seminars. So welcome, Dr. Ganguly. Okay, we are ready to hear you. Please.
Thank you, Dr. Sarkar, for your kind introduction. And first of all, I want to apologize for being late. I was having some technical issues. Hopefully it's all sorted and it should be fine. Before I begin, I want to thank Father Principal Sir and all the faculties and students of the Department of Microbiology and the Department of Biotechnology for giving me an opportunity to talk about my research in this platform here today. Very special thanks to Dr. Is the reason, I mean, he is one of the reasons why I pursued this career in biological research. He was extremely motivating in my plus 12, and a lot of thanks to you, sir. Okay, so I'll share my screen here. Let me see. Can you see this? Can you see the screen?
Yes, absolutely.
Okay, great. So as Dr. Kostri was mentioning, so my research is mostly has been on fibrosis. During my PhD, I studied cigarette smoke induced emphysema and pulmonary vascular fibrosis. But I showed that in the alveolar region, due to cigarette smoke, there is an extensive damage of the alveoli, which you can see here. There's an increase in these air spaces. However, at the vascular region, a very opposite thing happened. Whereas in the alveolar region, there is a damage. There is a destruction of the alveoli. In the vascular region, the exact opposite thing happens, and there is a rebuilding phenotype. So fibrotic building occurs over here, and in the perivascular region, there is extensive fibrosis. During my postdoc, I am studying obesity and NASH associated liver fibrosis, where I see that due to, I'll mention it, unhealthy diet over here right now. I'm going to explain it later. Due to unhealthy diet, mice and human, they develop extensive fibrosis in the liver. So in general, a unified mechanism of fibrosis in different organs, it seems that every time it starts with a damage, then there is an inflammation. Body tries to resolve this inflammation, and in some cases, if the damage is persistent, then there is a derailment of this resolution phenotype. Something goes wrong, and then chronic fibrosis occurs. The main manifestation of fibrosis is deposition of extracellular matrix, in particular collagen. So today, I'm mostly going to talk about my research involving liver fibrosis, which I conduct during my postdoc tenure. I always like to begin with this slide. It's kind of a reminder to myself and everybody that we are in an obesity pandemic. And in a report which is published very recently by New England Journal of Medicine, it says that by 2030, nearly 50% of adults will be obese and 25% will be severely obese. And there are a lot of disorders which are associated with this obesity. One of them is called Nafld, or non-alcoholic fatty liver disease. Now, non-alcoholic fatty liver disease, or Nafld, it's a spectrum of disease, and I'll take you through this. So this unhealthy liver, again, due to unhealthy diet, unhealthy eating habits, lipid droplets starts accumulating within the hepatocytes. Hepatocytes are the major parenchyma cells within the liver. At the first stage in Nafld, lipid droplets start accumulating within the hepatocytes, and these white things that you see here in this cartoon, these are the lipid droplets. At this stage, it is known as simple steatosis. This is a very benign stage. It does not have much of pathophysiological manifestation, and doctors and scientists are not really much worried about this stage. Most of the people will have simple steatosis, will have accumulation of lipid droplets within their liver at some point in their lives. However, around 20 to 25% of simple steatosis patients, they progress to a more severe form, which is known as NASH, or non-alcoholic steatohepatitis, where this accumulation of lipid droplet is associated with severe inflammation. Some of these NASH patients progress further to fibrosis and cirrhosis. Cirrhosis is a more severe form of fibrosis where you find more intense collagen deposition. And some of these liver fibrosis and cirrhosis patients will eventually go into HCC, or hepatocellular carcinoma, or liver cancer. So as I told, simple steatosis is a benign form with not much of pathophysiological manifestation. Most of the research in our lab and in all over the world, it is directed at this stage. What makes a simple steatotic liver to progress to NASH? What are the factors? It's not known. And according to WHO or according to FDA, all the clinical trials, they are focusing on finding a molecule to prevent this transfer. People are okay with simple. We need to know how to stop the progression from simple steatosis to NASH. Very similarly, how to stop the progression from NASH to fibrosis. Again, how to stop the progression from fibrosis to HCC is the major focus of research in this field. Now, in any biological research or in any disease related research, a disease is a multifactorial thing. There are so many aspects to it, and this is more so in NASH fibrosis, because although the main manifestation is in liver, a number of other organs are involved in the disease development and progression. Also, there are different types of experimental research that we do with this aim of understanding the disease biology, understanding how the disease is progressing, and trying to identify suitable interventions to halt the progression of that disease. For example, we do descriptive research, we do research that involves target identification, functional validation of the target, and then we do several intervention research for identifying proper drug molecules in order to halt the progression. And of course, which is one of in the lab, that is mechanistic research. Now, each one of us researcher, we are like one of these guys, looking at one particular aspect of the disease. It is humanly impossible to look at the holistic view for one single person. That is why a very important term exists in biological research. I'm sure we all know this. It is collaboration. And I myself, I have been very fortunate because we have a very extensive collaboration with different academic groups throughout the world, and also non-academic, a number of industries, where each of our collaborators study various aspects of Nash and liver fibrosis. I myself, I specifically look at the immune system aspect of it, but however, I am very actively involved in the other aspects as well. Today, for my talk, what I'll try to do, I'll try to shed light on how we can integrate all these different experimental approaches and all these different aspects of Nash fibrosis, again, in order to understand how the disease progresses and in order to develop suitable therapeutic interventions. Okay, so the first thing first, we need a preclinical model for studying Nash fibrosis. We cannot study in human, we know that. We need a proper animal model for that. Unfortunately, one of the major reasons why there is not a single FDA approved therapy for Nash and liver cancer is that we actually do not have a proper preclinical model for studying Nash. All the established model had some or other limitations. So our first research aim was to develop an animal model for Nash fibrosis and to study the disease in that model. So this is a part of that descriptive research that I was talking about. Number one is identify and establish or characterize an animal model to study Nash fibrosis. And number two is to study the entire disease progression in that model. So in the first part of my talk, I'll talk about this descriptive research. Now, in order to characterize or develop an animal model, we made use of a mice which was developed way back in Australia. So these mice, they lacked a gene called ALMS1. This gene is somehow, we don't know how, but this gene is somehow related to eating. It is somehow related to food intake. So much so that when this gene is deleted, these ALMS1 knockout animals, they eat more. So these animals are known as FOS mice. It came from the fat Aussie mice. So if you see this data here, the red one, they are the FOS mice, and the blue one are the normal wild type mice. And as you can see, the FOS mice, they eat considerably more than the normal wild type mice. So these mice, by nature, they comply with one of the aspects of human obesity, that is overeating. The second aspect of human obesity is eating unhealthy food. So what we did, we took these mice and we exposed them to a diet which is called Western diet. Now, it is called Western diet because this kind of unhealthy diet was initiated in the West. But I know we Indians, we are also very much exposed to this kind of diet. This is the composition of this diet. It has high fat. It has around 20% fat, moderate amount of cholesterol, but a very high amount of sucrose. Now, when these FOS mice had this kind of a diet, so on one hand, we have overeating mice, which eats more like we all do these days, like the human do. And on the other hand, they are taking a not so healthy diet. What happens is that they very quickly, within 1 or 2 weeks, they develop obesity, they develop type 2 diabetes, they develop several other complications of metabolic syndrome, including steatosis. I'll show you an image here. So this is how fat they get. So this is a wild type mice and this is that FOS mice. Both of them are on Western diet here. Both of them are having unhealthy diet, but because the FOS mice eat so much more, they become so fat. And this is the body weight. The red one here is again the FOS mice. You can see a substantial increase in body weight compared to all other controls. Now, very quickly, within 8 to 12 weeks, these mice, FOS mice on Western diet, they develop Nash and fibrosis. So this red stained portion over here, these are the fibrotic, these are the collagen bands. This is called a serious red stain. It specifically stains that collagen band here. Now, what is interesting is that after these mice develop Nash fibrosis, if you replace that Western diet with a steatosis gets reduced as well as the fibrosis gets reduced. We call this group as FOS regression group. As you can see here, these white patches, again, these are the lipid droplets. So this is a normal healthy liver with no lipid droplet. This is an extremely steatotic liver full of lipid droplets. And during regression, when we remove the Western diet and put them on a normal chow diet, a lot of these white droplets that vanishes. Similarly here, there is no red staining here, almost very little. These are just the structural collagen molecules. During Nash at 12 weeks, there is an extensive deposition of collagen, these red stains here. During regression, most of this collagen vanishes. However, if instead of putting these these FOS mice to a normal chow diet, if they are continued on Western diet, very soon at around 24 weeks, they start developing HCC. So these are the tumor nodules here, as you can clearly see here. So basically within 8 to 12 weeks, these mice develop Nash fibrosis, and in around 24 weeks, they develop cirrhosis and hepatocellular carcinoma. As I mentioned before, Nash is also associated with several extra hepatic complications. We also found that at around 12 weeks, these mice also develop chronic kidney disease and also cardiac dysfunction. Overall, once we developed these mice, we made a very comprehensive study of the hepatic and extrahepatic alterations that happen during disease progression in these mice. Again, it's all a part of a descriptive experimental approach to study Nash. I am not going to bore you showing all the data about it, but I can take you through a brief tour about what we found in these mice. It's recently published. I'm not going through the data, but just to show you what we find is that these FOS mice on Western diet within 1 to 2 weeks, as I showed, they develop hyperglycemia, steatosis, a lot of oxidonitrosative stress and inflammation. Along with these changes in the liver, we have a collaboration with a gut group which extensively looks at the intestine. What they found that at this initial aspect, it is within 1 or 2 weeks. It is long before fibrosis develops. What they found that already there are changes also in the intestine. They found that there is an increase in intestinal leakage. That means this intestinal barrier, that it gets disrupted. And there is a lot of intestinal inflammation and as well. One consequence of disruption of intestinal barrier is that bacteria, they start leaking from the intestinal lumen into the portal vein. And what it does is that it brings this bacteria and the bacterial LPS into the liver. And that really augments the inflammation to a huge extent. Now, one effect of such kind of research in the gut is that they found that there is a microbial dysbiosis that happens at this stage. I'm not an expert on this, but what I know, what they reported is that there is an alteration of the ratio of intestinal microbiota. There is a change in the ratio of firmicutes to bacteroids. One therapeutic intervention that they're trying to do is that they're trying to take Nash mice and they're trying to reverse this ratio by treating these mice with with these microbes and to see if changing this reverse ratio can stop the disease progression. I just wanted to mention this because you guys in microbiome, you might be interested in this because research on microbiome, it's a very new and booming field these days, especially in the context of Nash. Okay, so as the disease progresses, we find that in 8 to 12 weeks, they develop obesity, dyslipidemia, and a lot of other changes along with Nash and fibrosis. And as I mentioned, they also should develop a chronic kidney disease. At this stage, if we give them chow diet, fibrosis reverses. But if they continue on Western diet, they very quickly develop HCC, they develop cirrhosis. At the same time, they develop cardiac dysfunction. So much so that majority of these mice we see, they actually die by around 32 weeks of age. One very interesting aspect that we found during the progression of Nash in these mice is that there is a change in the cytokine profile. There is a change in the inflammation profile in these mice. At around 1 or 2 weeks, some of the cytokines get upregulated, which really at the Nash stage, these upregulated cytokines, they take a dip, they come down, and a very different repertoire of cytokines, they start expressing at this stage. What is the function, how this cytokine and the inflammation govern this Nash we are studying that right now. So moving forward, an acid test are really a litmus test of how good is your animal model for preclinical study is when you compare your model with a human Nash model. So this is such a comparison where we took our Nash mice, we isolated all the RNA from the liver of that Nash mice, and we did a bulk RNA sequencing. We then compared the Nash gene signatures, the genes that are upregulated or downregulated in Nash in that mice with the genes that are already known to be upregulated or downregulated in human Nash fibrosis. And this is the data. As you can see here, it shows a very strong concordance between mice Nash and a human Nash, which means that the genes that are upregulated in mice Nash fibrosis, the same genes are also upregulated in human Nash fibrosis. Similarly, the genes that are downregulated in mice Nash fibrosis, the same genes are also downregulated in human Nash. This kind of research, first of all, it helps in establishing that you have a good preclinical model, but along with that, this is also very important for target identification. I'll come to that a little later. Switching gears, the second thing that I wanted to talk about is it's again a part of a descriptive experimental study for understanding Nash, but this is a very different approach. So here, what we are trying to do, we are trying to understand what are the changes that the hepatic cell types undergo during Nash. So we are trying to understand Nash fibrosis induced alterations in liver cells. And we do this at a single cell level. We do a single cell RNA sequencing. We do single nucleus attack-seq for this. So just to begin with, there are several types of cells that are present in the liver. The hepatocytes, they form the bulk of liver. 80% of liver are composed of these hepatocytes, which are the major parenchymal cells. This apart, there are the stellate cells, which are the major myofibroblasts that secrete fibrosis. I'll talk about stellate cells later. And then we have the liver resident macrophages, which are the Kupffer cells, and also the recruited macrophages that come from the bone marrow. And of course, we have the endothelial cells. So each of these cell types, two things happen. They undergo changes during fibrosis progression, and also they talk to each other. For example, what is known about the cell-cell interactions during Nash in liver is that, First of all, by now we know that NASH fibrosis begins with accumulation of lipid droplets in the hepatocytes. So these are the hepatocytes and these are the lipid droplets inside the hepatocytes. The accumulation of these lipid droplets, it kind of, if the limit exceeds, it kind of bursts out the hepatocytes, you know. So, sorry. So the hepatocytes kind of burst open and releases all of their internal content into the liver. That triggers the activation of immune cells. Immune cells activate these myofibroblasts, the hepatic stellate cells, which are the ones that secrete collagen, causing fibrosis. So these cells, they themselves undergo changes and then they talk to each other. We use single cell sequencing platform in order to understand the changes that each of these cell types undergo during NASH. Just a very brief note about single cell RNA sequencing. So I just showed you the result of a bulk RNA sequencing. So in bulk RNA sequencing, what we do, we take the liver tissue, we isolate all the RNA from that tissue, and that RNA we use for sequencing. We have one control tissue and a NASH fibrotic tissue. So we get the control RNA, we get the NASH RNA, and then we compare the RNA sequence from those two. Now, what happens in that case is that, as I showed you, liver consists of different cell types. So what we are essentially getting here, these genes that we get out of this bulk RNA sequencing, we do not know where these genes are coming from, whether they're coming from the hepatocytes or they're coming from the macrophages or they're coming from the stellate cells. So the individual cellular signal is getting lost here. For single cell analysis, what we do, the first step is we isolate all these cells. We isolate the hepatocytes, we isolate the stellate cells, we isolate the macrophages, and then each of these cell types separately undergo sequencing. So each cell here will have a very distinct expression profile. And what we get a cellular heterogeneity, and the changes here are revealed, which gets masked in case of bulk RNA sequencing. But still, bulk RNA sequencing is very important because in single cell, we see the changes in individual cells, but we lose the interactions between them. That we can actually study in bulk RNA sequencing. I mean, it just depends on our final aim, whether we are going to go for bulk RNA sequencing or single cell sequencing. Anyways, so in order to study the changes associated with each cells during NASH, we employ this technique of single cell sequencing. The first data, I think the main data I'm going to show today is refers to the changes in the stellate cells. So as I told, hepatic stellate cells, these are the main collagen secreting myofibroblasts. So these are in a normal liver, these stellate cells, they remain in a quiescent form. They are not activated. All they do is they store vitamin C. Vitamin A, sorry. Now, once the liver is injured, once they get some signals from the macrophages or the damaged hepatocytes or the endothelial cells, these quiescent stellate cells, they become activated and they start secreting collagen. So this is the single cell RNA sequencing of the hepatic stellate cells. Now, if you see here, we took three conditions. So this FOS week 0, this is the control. So these are at the 0 week, they have not been exposed to the Western diet. Now FOS week 12 is the mice which develop NASH fibrosis. So what we expect here is that FOS week 0 will be normal. So they will have these quiescent stellate cells. FOS week 12 will be fibrotic. So they will have these activated stellate cells. And then we have this regression group. If you remember, regression group is the group where after these FOS mice on Western diet, after they develop NASH, we switch them to a normal chow. And then in these mice, what happens? Fibrosis regresses. So there's a reversal of fibrosis. So this regression group is the recovered group. Now, so these are the different clusters of the stellate cells. What we do once the stellate cells are isolated and sequenced, bioinformatically using softwares, they are plotted in maps called the UMAP. And the cells which are similar in their nature, which express similar kind of genes, they are clustered together. There are a number of known marker genes for the quiescent stellate cells, and there are a number of known marker genes for activated stellate cells. I'm going to talk about 4 marker genes here. The first one I'm going to, so these are, there is a gene called LRAT and there is a gene called desmin. Each of the stellate cells, regardless of whether they are quiescent or activated, they are going to express these 2 genes. That's what you see here. If you see the expression profile here, yellow shows, yellow is very high expression and green is kind of moderate expression and purple is very low expression. So if you see here, LRAT is expressed in all these clusters because this proves that what we have isolated are actually stellate cells. It's important to prove that as well because this isolation process is a very tedious process and frequently you find impurities and contaminations from other cells. Similarly, for desmin, you can see a moderate amount of desmin expression in all the clusters. Now coming to the more interesting one. This is a Col1 alpha 1 gene. This is basically the collagen gene. The collagen that is secreted by these stellate cells, this is the gene which governs that protein. There is another gene called ACTA2. Both this Col1 alpha 1 and ACTA2, they are found exclusively in activated stellate cells. Now, if you see the expression of Col1 alpha 1 and ACTA2, you see that they are mostly in this cluster 5. And they're absolutely absent in this cluster 2 and 0, and their maximum in this cluster 5, 9, little bit over here, a part of this cluster 4. Based on this, we can identify that this cluster over here, this population, these are the quiescent cells. And indeed, they come from the FOS weak 0 mice, which is the normal mice. Again, based on the Col1 alpha 1 and ACTA2 expression, we find that this A1 and A5 cluster, these are the activated stellate cells, and they come from the FOS12 weak group, which we know these are the Nash group, which we know that they will have this activated stellate cells. Now, these are all known. The novelty here, the surprising thing here was that we found that there is one cluster, there's actually 2 clusters over here that express LRAT and desmin, again proving that these are the stellate cells. However, they do not express ACTA, which is a marker of activated stellate cell. And they also express very low amount of Col1 alpha 1. So this population was never found before. So we hypothesized that this can be, an intermediate form, which are really not quiescent, not activated, and kind of intermediate form. And we found that both of these clusters exclusively come from the recovered mice. So we wanted to study these clusters in more detail. I am skipping the data for characterizing this cluster, but what we found that this new cluster, they are a population of stellate cell, which we have named them as inactivated stellate cells. What happens? They are derived from the activated stellate cells. So they were once activated, but now during regression, they have lost their activation status. But they do not go to that quiescent form. They are at an intermediate state. If the insult comes again, they get very quickly activated. So we characterize this cluster, and we found that they express several unique genes, which is not expressed in any other stellate cells or any other cells. One such unique gene is GABRA3. So whatever we found by single cell sequencing, by bioinformatic analysis, the general rule that is that you have to establish that in vivo model. So this is, so till here, we used the single cell platform, we used bioinformatic analysis and all. Here we have, here in this data, we are trying to establish what we got through bioinformatics in an in vivo model. So this is then immunohistochemistry, that is the immunostaining of this GABRA3. If you see this GABRA3, the FOS 0 week is, again, it contains the quiescent stellate cells. FOS plus Western at 12 week, it contains the activated stellate cells. In both of them, you see there is no positive staining of GABRA3. Only in this FOS recreation, which contains this inactivated stellate cell, we see this positive staining of GABRA3, which establishes that FOS regression, during regression, there is a population of, there's a very unique population of stellate cells that comes up, and they express some unique markers like GABRA3. So now the thing is that, so what? We have identified a new class of stellate cells, which was not known before. We have characterized them. We found a new marker for them. But how does that affect in therapeutics? I mean, that's our aim, right? We want to know more about the disease, but ultimately we want to solve the problem. We want to treat the disease. So based on this knowledge, what we are trying to do right now is we are taking activated stellate cells. This is a research in progression. I don't have a data to show you, but I can just tell you the theme of our research over here. So we are taking activated stellate cells and we are that stellate cell. So we are forcefully expressing GABRA gene inside these activated stellate cells, and we are trying to see that those stellate cells, those activated stellate cells that will get that GABRA gene, will they spontaneously become inactivated despite the insult? If they do, then we can say that this GABRA3 gene can drag down, can kind of transform an activated stellate cells into an inactivated one. If we can show that, then we can find some therapeutic application of this GABRA3. Okay, so that is, as I told, that's very much a research under progress. As I talk, that is what is going on in our lab right now. So moving forward, okay, before I move forward, I just wanted to tell you something as a word of caution. Right now here, for every evidence, I'm showing you one data. I'm mostly showing you the immunohistochemistry or the histology data, but I just want to take this with caution, is that for everything, for every evidence, one data is never sufficient. I do not want to bombard you with this data. That's why I have selected one. But normally we have a panel of experimental procedures that we do in order to establish one evidence. For example, we mostly do a lot of qPCRs, qRT-PCRs, and then we do a lot of Western blot for the protein level. So for all these that I am showing you, there is a qPCR data, there is a Western blot data, and there are more additional staining data. So for everything, we have to repeat the same experimental outcome in multiple data and multiple evidence in order to confirm it. Okay, moving forward, the third aspect, as I talked about target identification, and I told that this kind of bulk RNA seq results, and especially comparing mouse NASH with human NASH, along with establishing how good your mice model is, it also helps you to identify proper targets for the disease. So here is one target. So it's like very simple and it's very straightforward. These genes that are upregulated in both human NASH and FOS NASH, any of them can be a very useful target. So we chose one of them, which is called TREM2. This is one gene that is upregulated both in human NASH and mice NASH. So we thought that if it is so, if we knock down or knock out this TREM2, is there a difference? Can we reduce NASH. So for that, okay, a little bit about this TREM2 molecule. So not much is known about this TREM2 protein, but it's just that these TREM proteins, they belong to immunoglobulin superfamily. These are the cell surface receptors, and they are primarily lipid sensing receptors. They bind to various lipids. They also bind to LPS. Nothing much is known about the role of TREM2 is NASH, which shows that this can be a very novel target. However, it is known that a loss of function mutation in TREM2 is a risk factor for Alzheimer's, but not much is known in NASH. So next, what we tried to find out is that what is the role of TREM2 in NASH fibrosis? But again, before going into that, we need to establish what we find in this RNA-seq. RNA-seq it's just one technique. It can go wrong. So the first thing is that we need to show that in mice model and in human NASH patient, TREM2 is indeed upregulated. So this first panel of data shows that I'm not going to go into details, but this first data, this is the TREM2 mRNA. This is the TREM2 relative expression. And as you can see here, this is the FOS plus Western diet 12-week. This is the group that develops NASH. As you can see, compared to control, which is the 0-week, there is an increase in TREM2 expression. And during regression, the TREM2 goes down. So basically, with the increase in fibrosis, TREM2 expression is increasing. With the reduction of fibrosis, the TREM2 expression is going down. The same thing we also established in a couple of other NASH models. These were the already previously established NASH model. We also showed that this TREM2 change is also, we can also see that change in other NASH models, just to prove that this is not one model specific thing. This is a more universal thing. And then this is a human NASH patient. So this lower panel is in human NASH, and this red one, again, the red one is the high TREM2 expressing population. So as you can see, compared to healthy obese, there are more red in NASH, which shows that TREM2 expression is increased in human NASH as well. This data, it's in another cohort of human patients. We show that the TREM2 expression, the gene expression of TREM2 is increased in when the fibrosis increases. So F0 and F1, these are the clinical fibrosis scores. When a patient comes to a clinician, based on the amount of fibrosis in the liver, they are given various scores. F0 is definitely the control, healthy. Actually, it's not healthy, but it's a non-fibrotic liver. Based on the degree of fibrosis, they get a fibrosis score of 1 or more than 1. So as you can see here, as the fibrosis score increases, there is an increased amount of TREM2 transcripts. And this final figure here, this shows that this is a healthy human. Compared to a healthy human, there are more number of TREM2 positive cells. Each of these green circle here, they represent TREM2 positive cells. One interesting finding is that, okay, and this red one is the collagen. One interesting finding for us in human patient is that as the fibrosis stage increases, as more collagen is being deposited, these TREM2 cells, they tend to cluster in that damaged fibrotic area. So if you see this confocal microscopy figure, this green, the green positive TREM2 cells, they are mostly clustered along with that collagen fibers. Now, the next thing we wanted to find out that which cell types in the liver upregulate TREM2. Here again, we go back to our single cell RNA-seq. The previous single cell RNA-seq that I showed, that was done with the hepatic stellate cells. Now this single cell RNA-seq are done mostly with the macrophages and other immune cells. So these are all the liver NPCs. NPCs means the non-parenchymal cells. So as I told that the hepatocytes represent the liver parenchymal cells, all other cells except hepatocytes, they represent the non-parenchymal cells. So here, these are all the cells except hepatocytes, and we have also taken the stellate cells apart. So all the liver cells except hepatocytes and the stellate cells. So if you see here, these are the various types of cells. These are the recruited macrophages. These are the resident macrophages. If you see the TREM2 expression, is the high TREM2 expressing cells, and the yellow one are the cells which do not show any TREM2 expression. So as you can see here, TREM2 expression is specific to the recruited and resident macrophages. Moving forward, we used another technique. I'm not going to describe this. I'll just brush through this. We used another technique which is called the single nuclear RNA-seq. So in this single nuclear RNA-seq, instead of isolating the cells, we take out the nucleus of the cells, and then we do the sequencing. We modified this technique a little bit, and we performed what is known as single nuclear attack-seq. With attack-seq, what we try to find the open chromatin regions in the DNA. By open chromatin region means that region of DNA which is actively transcribed and translated, that will be open, that will be free of the histone proteins, right? So if the open chromatin regions in the DNA will be the part of DNA which is most actively expressed. Now I'll just go through this data real quick. So we took all the nucleus from the liver, and then we can plot them and differentiate them into different population of cells in the liver. And so these are the endothelial cells, fibroblasts, hepatocytes, macrophages, and all. And what we see here is that the TREM2 locus is open only in the macrophages. That means only in the macrophages, only in the DNA that is within the macrophages, there is an open chromatin for TREM2, which proves that this TREM2 is only expressed in macrophages. Now, coming to the most important thing, what is the role of TREM2 in NASH? So the best way to find it is if we use a mice where TREM2 is deleted, and we see the effect of that deletion on NASH development. So we took the phosphos mice again, and we took a phosphos and TREM2 knockout mice. So this phosphos and TREM2 knockout mice is a double knockout mice which lacks the ALMS1 gene, which makes them phosphos, and also the TREM2 gene. What we expected that since the TREM2 expression increases with NASH, if we reduce TREM2 expression, probably the NASH will reduce. Now, this is the data. This is again the histology data. So if you see here, this is the phos mice and this is the TREM2 knockout mice. Contrary to our expectation, as you can see, there is a much more intense fibrosis in the absence of TREM2. Again, just to remind you, the red one, these are the serious red stain sections, and each of these red one are the collagen deposits. So there is a huge increase in deposition in the absence of TREM2, which shows that TREM2 is actually a protective molecule. Now, this is again the Col1 alpha 1 gene expression, which says the same thing. We validated our findings in yet another one NASH model. Again, I'm not going through the details, but as you can see, compared to wild type, the TREM2 knockout mice, there is a more extensive fibrosis. In fact, what we find that these TREM2 knockout mice very spontaneously developed tumor. So overall, our conclusion from here is that TREM2 is it's an important target. It's still an important target for NASH because TREM2 deletion, it is worsening the disease. So right now, what we are doing in collaboration with a pharmaceutical company, we are trying to find out ways where we can increase the TREM2 expression, where we can increase the TREM2 protein in NASH, and to see that if that can prevent NASH. Moving forward, I'll come to the last part of my talk. So this same data we can use, by now you know that we can use this kind of studies to identify targets. We identified yet another target, which is known as UBD. I'll rush through this. So again, we found that UBD is upregulated in both mice and human. What is interesting was that there is nothing much known about this protein UBD. And we found that, again, from single cell RNA sequencing, we found that UBD is expressed mostly in the hepatocytes. I don't know if you can see the color over here, but just trust me, UBD expression is high, mostly in the cluster 3 and 15, which are the hepatocytes. So by now, we found that GABRA is expressed on the stellate cells. We found that the TREM2 is expressed in the macrophages. And now we find another protein, another target, which is now expressed in the hepatocytes. Now, again, just following our routine intervention, we want to see if we knock down or knock out UBD, there is any change in Nash fibrosis. Now, there is a very interesting thing here. The presence of UBD only in the hepatocytes gives us a very unique advantage. I'll talk a little bit about this. There's a pharmaceutical called Ionis Pharmaceutical, which have developed some antisense oligonucleotides. Now, these antisense oligonucleotides, they can specifically target one particular gene and can silence that gene. The beauty about these antisense oligonucleotides is that if you tag this antisense oligonucleotide with a particular linker, which is called GalNAc, they are specifically taken up by hepatocytes. So it becomes a very target-specific approach. So what we did, we took these, again, NASH mice, then we treated them with these antisense oligonucleotides. We just injected them. So there is a very good advantage of this antisense oligonucleotide compared to a knockout model, is that when you get a human NASH patient, you cannot knock out any gene in that patient, right? So knockout models are very good to understand the disease, but they have limitations in terms of therapeutic applications. But what these specific oligonucleotides give, the advantage that they give is that if there is a NASH patient and if you have a therapeutic antisense oligonucleotide in your hand, you can easily inject that patient with this, as like you do in case of most drugs, and you can easily treat that patient. That is exactly that we do, not in patients, but in mice. We injected NASH mice with this antisense oligonucleotide intraperitoneally, and then we checked the effect. As you can see here, so these are the various antisense oligonucleotides that we used. Antisense oligonucleotide 1, 2, 3 is the control one. So this is basically a scrambled oligonucleotide, which is not specific to anything. Okay, it's just a sham control to take care of the injections and all those things. So this is our control, ASO133, and these three are our different antisense oligonucleotides which are specific to UBD. So as you can see, for all these antisense oligonucleotides that are targeted to UBD, they are capable of knocking down the UBD gene expression to a different extent, but at least they successfully knock down UBD. While knocking down UBD, what we see that the Col1 alpha-1 gene expression also comes down, which is the major readout, which shows that if we knock down UBD, the fibrosis actually comes down, which is also seen here in, again, another serious red staining, that if you see here, this is ASO133, which is the depositions here. But when you treat with ASO615, which is an antisense oligonucleotide that is specific to UBD, you can actually reduce the fibrosis in these mice. So overall, in contrast to what we see in TREM2, we see that knocking down of UBD actually improves NASH. So overall, as a summary, what I discussed today is that we use different approaches. The aim is to study the disease progression of NASH fibrosis and to develop different intervention means to stop the disease, to cure the disease. As a descriptive study, we have characterized a preclinical animal model for studying NASH. And in that same model, we have followed the kinetics of hepatic and extrahepatic alteration during NASH. And we have also characterized in detail the phenotypic changes of hepatic stellate cells in this FOS model. And we have identified a population of inactivated stellate cells during Nash regression. How we transfer this descriptive science into a therapeutic one, which is our major goal, it's under process in our lab. The second part of experimentation is the experiments that are involved with target identification. We have identified 2 potential targets for Nash, and then we have done intervention studies. We have shown that the inhibition of TREM2 worsens Nash fibrosis HCC, and right now we are trying to develop techniques by which we can augment TREM2 expression in Nash mice so that Nash can be resolved. At the same time, just in contrast to TREM2, we show that the inhibition of UBD actually improves Nash. So right now we have these antisense oligonucleotides in our hand, which can be used as a therapeutic approach for Nash fibrosis. And we will start a phase 1 clinical trial very soon with these antisense oligonucleotides. We have also used, I have skipped this part, but we have also used this FOS model for different drug screening. For the last 1.5 years, we have been getting drugs which are in pipeline for in different pharmaceutical industry, and we treat these mice with those drugs to see that if there is any reduction of fibrosis. Also, part of our research, and it's a very important one, we also need to identify biomarkers. We have already identified a reliable plasma biomarker for Nash HCC. This is under patent consideration, so I could not discuss this much, but it's also actually a very, very interesting aspect of our collaborative research. And finally, we are undergoing mechanistic studies. This is my favorite part. It feels so good when you dissect out each of the puzzles. What is the mechanism of Nash? How is actually absent? We know that the absence of TREM2 worsens Nash, but how it happens? We know that the absence of TREM2 it improves. We know that the absence of UBD improves Nash, but how it happens, we don't know it. We don't know yet. It's under experimentation in our lab. So finally, I want to thank my PhD supervisor, Professor Kaustubh Panda, and Professor IB Chatterjee for training me. I owe them a lot. And my current postdoc mentors, Dr. Devanjan Dhar, Dr. David Brainerd, and Dr. and all the members of our lab. So Nas and Ashra, they are the graduate students. They work very closely with me. And Shao and Naidika, Shao especially is an expert in isolating cells and establishing primary culture. So he takes care of most of those cell isolation part. And then, as I told, we have a lot of collaboration, and I'm thankful for all of them because I get an opportunity to look at the whole Nash fibrosis aspect because of their help. So yeah, finally, thank you for your time, and I can take up any questions as it comes. Yeah, I'll stop the screen sharing here.
Thank you, Dr. Ganguly, for your wonderful presentation. It was a complex story, starting from Nash induction to revealing the underlying mechanisms and finally treatment option. Okay, it's really wonderful. And we have a question here. What healthy diet should we follow to keep our liver and kidneys healthy?
So as you saw, this Western diet, it has actually moderate amount of cholesterol and a lot of fructose. So in the Western world, there are things called corn syrup. There are some high fructose corn syrup, which are really bad. For us, I think if we keep on taking high fiber diet and stuffs, they will be good. The fast food are really, really, really not good. And we are into that lifestyle these days. I don't know what is the solution, how we stop that. But I would say, to reduce the fast food intake as much as. I'm not a dietitian, but what I see that in my model, this fast food, especially the high fructose diet, is really bad. I try to do that. I'm not always successful, but that's why I keep reminding myself with that first slide. Yes. But yeah, it's good to avoid the first.
Website you used in the induction of NaSH, I noticed that it has 20% of fat and 33% of sugar, if I'm right.
Right.
So which one is more harmful, sugar or fat?
So basically, initially we thought that we all know that the cholesterol is the major component, but now what recent research is showing that it's not the cholesterol, even more than cholesterol, it is the fructose. So what fructose does, fructose induces uncontrolled de novo lipogenesis. So the de novo lipogenesis, which starts from sugar, if you see, it had a feedback control mechanism, it stops. But fructose, it induces uncontrolled de novo lipogenesis. So there's no feedback inhibition there. So that's why excessive taking of fructose is more of a culprit. So if we made a head-to-head comparison, a diet which is more in fat and a diet which is more in fructose, we saw that the more damage is done in the diet which is more in fructose. So there is a specific diet which is called high-fat diet. That is the diet which is more in fat, and this Western diet is more in fructose. Compared to high-fat diet, Western diet causes much more intense damage. You can see that many foods it is labeled as it contains high fructose corn syrup. That is extremely damaging. I mean, especially in US, people have they take pancake with syrup. That syrup is loaded with high fructose corn syrup. That's very bad.
It provides instant energy, but ultimately, detrimental to our health.
Because it induces the de novo lipogenesis, and what happens, even if you don't take fat from outside, de novo lipogenesis is going to overload your cells with fat. And when the capacity of adipose tissue to store that fat, when it exceeds, it comes to liver. And DNL will anyway, it will take place in liver, and the hepatocytes get filled up with fat.
Thank you. Extremely important work you are carrying on. And all these factors, TREM2, GABRA, then UBD, you have identified all the crucial points in all the cells in our liver. It's really very nice to hear you, sir.
Thank you
so
so much.
Thank you. Yes, thank you. Thank you for joining, and thank you for accepting our invitation. Thank you so much.
Thank you.
It's unmuted. Yes. Thank you, Dr. Shorodita Ganguly. And now I would like to request PS ma'am to present the plaque.
So this is for you, Dr. Ganguly.
Thank you so much. It's
from Department of Microbiology, Xavier College.
Thank you so much.
Hope to meet you in offline mode.
Absolutely. Sometimes soon. I love
really, I would like to thank Dr. Ganguly for accepting our invitation at a very short notice. We are really grateful to you. Thank you very much.
Sir, thank you so much.
It is a lot of motivating instances that our own old students, they have gone so far in research. Makes me proud. Thank you.
Thank you. It's always great to meet you and talk to you.
Thank you very much.
Thank you.
Thank you.
I would like to thank our first speaker for the day, Dr. Shorodita Ganguly, for delivering such an insightful talk to our audience and our moderator, Dr. Kosturi Sarkar ma'am, for wonderfully conducting our first technical session. Now, we would proceed on with the second technical session of the Frontiers in Biological Sciences, Chapter 5. I would like to call upon our moderator for this session, Professor Dr. Mahashweta Mitra Ghosh, ma'am, to introduce our second speaker for the day, Dr. Shreyoshri Chatterjee, a postdoctoral scholar from the Scripps Research Institute, USA. She would be delivering a presentation on the topic, Role of Erusamide in Retinal Homeostasis. Over to you, MMG ma'am.
Thank you, Sanjita.
Thank you, ma'am.
It's my pride. It's a proud privilege for me to welcome Dr. Shreyoshri Chatterjee. Right, she actually did her graduation from our own college, and after that she did her master's from Presidency College, and she pursued her completed her PhD from Iowa State University, USA, and her topic of research was synthesis and conformational analysis of 2266 tetramethyl bipyridyl masked 1,2-diols and its application in the synthesis of oxylipins from Dracosium florets. Okay, so after that she joined as a, right now she is a staff scientist in Scripps Research Institute as a postdoctoral research associate. And within this very short span of time, she has published many papers. And without taking much of time, I would like to highlight on the topic on which she will be discussing today is the role of erucamide in retinal homeostasis. So my heartiest welcome to Shreyoshri Chatterjee. Over to you.
Can you guys hear me?
Yes, we can hear you.
Can you guys see my screen?
Yes, your screen is visible.
All right. Good evening, everyone. And I'm Shreyoshri. I'm a postdoctoral scholar at the laboratory of Professor Dale Boger at Scripps Research Institute. And a very, very big thank you for inviting me and giving me an opportunity to share my research with you guys. It is always good to be back home, back where I actually started my journey as undergrad in the chemistry department, but I still did my pass for the subject was microbiology. So I was in close connect with the department all throughout my undergraduate days. And so as mentioned in the introduction, I am basically by training and also at heart a synthetic organic chemist. And I've always been interested in making molecules that has some biological function. So my PhD training was completely on making new molecules and studying those molecules. But during my postdoctoral research, I shifted my gears a little bit. And my focus of my research was more on the interface of chemistry and biology in the Boger lab. I've been a part of many projects that deal with both chemistry and biology, and I would like to share one of my ongoing projects in the Boger lab. So in this project, we study the role of a bioactive lipid, which is called erucamide, in eye physiology. This project is a highly collaborative project, which labs in the molecular biology department, chemistry department, chemical biology department, polymer chemistry department, all bring in their expertise together to solve the problem that, to find out the role of erucamide. And this project has been spanning for over 10 years. And it was unattended in the between for around 5 years. And back in beginning of 2019 is when me and my collaborator took up this project and started going on from there. So I'll just introduce the lipid that I'm talking about. So uricamide is a monounsaturated fatty acid amide, which has been found to be the most abundant metabolite in wild-type retinas. So my labs that we collaborate with, a molecular biology lab, they specializes in eye physiology and diseases, and they from one of their observations, they found that this metabolite to be highly expressed and also highly dysregulated in many models of retinal degeneration. So if you look at the structure of uricamide, which is figure 1, it is a long chain hydrocarbon, which makes it really breezy and not soluble in the buffers or water-based buffers that you have, which would make it amenable to inject into retinas or eyes of rodent molecules. So in order to come up with a good delivery system for these bioactive lipid, the collaboration with the polymer chemistry lab in the University of California, San Diego started, where they developed microparticles and nanoparticles which aid in the delivery of this lipid on to the retinas. So here is a section of the retina that shows that uricamide injected nanoparticles generally results in increased vascular permeability and inflammation. So as I told you, the project was remained unattended for a good 5 years. So before we joined the project, the mode of delivery of uricamide was microparticles. But my collaborator from the molecular biology lab, she tried to find a better method to deliver uricamide onto the retinas, and the collaboration with the polymer chemistry lab happened, and they came up with this nanoparticles, which proved to be a better system of delivery of this bioactive lipid. Now, it was also found that an optimal amount of uricamide was essential to maintain the retinal structure. A high amount of uricamide causes the causes inflammation, which is detrimental to the health of retina, and a low amount of uricamide results in thinning of the retina, which is also not a desired effect. So maintaining an optimal amount of uricamide in the retina is very important. But knowing all this, it was still unknown what is the mechanism of action of this bioactive lipid. So to know the mechanism of action, we had to know what the binding protein is, which is it the vascular endothelial growth factors or any other growth factors or cytokines? And in order to know what the binding protein is, we need to know which cells does uricamide target. So is it the RPE layer or the endothelial layer, or there are other targeting cells that can do it? Or there is a crosstalk between these various cellular layers that results in the desired bioactivity of this lipid. So here is where my lab, which is a purely synthetic organic chemistry lab, comes in. And in order to find out or answer these two questions, what is the binding protein and what is the cellular target, we design chemical probes. So these probes, which is generally the structure of these probes are similar to that of the original bioactive molecule, which is uricamide in this case, but you attach different tags to the probe, which will help in either imaging or other analytic studies, which I will go into further. But for finding out what the targeting cells are, we attached a fluorescent dye, which is called BODP, to visualize the uptake of uricamide into the cells. And to find out target proteins, we used a biochemical technique, which is called photoaffinity labeling. And I will come to it more when I discuss it in further details. So here is the structure of the fluorescent probe that we designed to find out what are the targeting cells of uricamide. So as I told you, I'm a synthetic chemist at heart. I will not want to bore you with a lot of chemistry and chemistry experiments, but I would like to walk you through the how we, synthetic chemists, look at a molecule, break the molecule into smaller pieces till we arrive at something that is commercially available, and then go back to the lab and start joining these molecules to get to the desired product. So this process of breaking these molecules is known as retrosynthetic analysis. So I'm just going to walk you through that, and no chemistry experiments. So what we do is we look at strategic bonds in the molecule that can be broken, form smaller fragment pieces, and can be joined with known chemical reactions. So if we look at this molecule, the bond here is this 5-membered ring, which can be formed by a reaction known as the Huygens cyclization or 1,3-dipolar cycloaddition, and which is the most common form of click chemistry. Applications of click chemistry and this cyclization you'll see further ahead as well. So this gives us these two fragments, which is this alkyne over here and the azide, which is attached to the BODP dimer. So this reaction or this experiment was more cleaner and surprisingly cleaner than I expected. But these dyes are extremely light sensitive. So when we are running these reactions, it has to be in absolute dark. So there's no lights on, and which might just cause a lot of unwanted accidents in the lab, like breaking glassware and stuff, but in a chemistry lab, that is least of our worries. So these dyes, the only upside of working with these dyes is that they are really colorful ones. And it's an absolute delight to see that color in rather boring organic chemistry labs. So then this alkyne fragment can be obtained from this ester via 2 steps. And this ester that we have, so again, the strategic bond that we break is this double bond, which can be ligated again to give us these 2 fragments that can be ligated again via a reaction known as the Wittig reaction, which is a very known reaction. And this reaction was also a little problematic for me because the intermediates in this reaction was, they decompose every time. And it took a lot of painstaking efforts to actually get this reaction work in a reasonable yield. So this fragment, which is called the phosphonium salt, can be prepared from starting material that is available commercially via 5-step. And this aldehyde, the other partner, can be obtained commercially. So, so with the dye-labeled analog in hand, we went ahead and injected these probes onto the retina. Although we introduced a pretty polar group with charged particles, but to our dismay, these particles were still not soluble in any water-based buffers that we had. We still had to resort to the use of nanoparticles as a mode of delivery of these analogs. So here we have the section of the retina that was viewed. So these 3 is just the enlarged version of this diagram. So if you see these red dots here are actually those corresponding to the dye labeled analog here. And if you see these green spots are due to the silicon nanoparticles that are used to deliver the probe. Now this is just these 2 images superimposed against each other. And you can see that both the green and the red spots, they appear at the same position. Which is a confirmation that the silicon nanoparticles are still a very good delivery system for an analog of fluoropamide, which structure is quite different from the original molecule. So then with this in hand, we incubated for a longer time, which is, I think the retinas are isolated after 3 days of injection. And then we tried to find out what the targeting cells actually are. So this is also an enlarged, zoomed in version of a retinal section. So these red stains that you can see, which is due to GS-lectin, which is a marker for endothelial cells. The green dots that you see is a marker is due to CDB11, which is a marker for macrophages or microglial cells. These are fluorescence due to the BODP dye. And this is all three merged together, the final image here. If you look closely, you can see that the green dots and the purple dots merge together completely, which hence we can conclude that the BODP-labeled europamide has been uptaken by macrophages or microglial cells. So with the targeting cells that was confirmed, now we next move to find out what the target proteins are. And that finding out the target proteins, we use a technique which is called photo-affinity labeling. It is one of the most commonly used methods to find targets for bioactive small molecules. What we do here is we synthesize an analog of the bioactive molecule, and which is very similar to the probe, but we attached something called a photophore, which upon excitation with UV light converts the binding of the ligand to the protein into reversible covalent binding. So it further strengthens the binding between the ligand and the protein. So, and after that, with the ligand protein conjugate, now we have a reporter group or reporter tag, which is attached as per our analysis needs. So how we want to analyze this conjugate of the ligand and the protein, accordingly, a reporter group is tagged onto the molecule. So the most commonly used photophores are aryl azides, benzophenone, and diazidine. So aryl azide, so when you shine light on aryl azides, they break down to form these nitrine intermediates here, which is very reactive. And when you have benzophenone here, they break down to form this dry radical, which is the reactive intermediate responsible for the further covalent bonding. And when you use diazidine-based photophores, it breaks down to form carbene, which is the reactive intermediate in the covalent binding. So with the various photophores, after forming the ligand, so in the probe, you can attach a reporter group that you want to use to analyze. But usually if you directly attach the reporter group, the resulting probe becomes very bulky and very cumbersome to analyze. And also it might interfere in the interaction of the ligand with the protein. So most commonly, the reporter group is added on or conjugated after the covalent binding event between the ligand and the protein. And the most commonly used reaction to tag along these reporter group is a click chemistry reaction, which is I had shown you before. It is also a copper catalyzed reaction between an alkyne and an azide, as you can see, to form this triazole intermediate. So you can put the alkyne either in the ligand or in your reporter group, or the azide also in the ligand or the reporter group, and then you can snap these two together with introduction of copper to finally get the conjugate that you want to study. Again, as I said, depending on how you want to analyze the conjugate, you can either introduce fluorophores like dyes or biotin or other radioactive label to analyze and study it further. Now, to give you a schematic idea of what happens in a photoaffinity labeling studies, so you take the ligand and then you incubate the ligand with the protein mixture for a given amount of time, and then you shine UV light on it. Now, the UV light generates a reactive intermediate. So what I'm showing here is a diazine-based molecule. So you lose nitrogen to form that carbene radical. Now, this carbene radical will insert onto the protein, insert onto carbon-hydrogen, nitrogen-hydrogen bonds, and form a covalent bond between the protein and the ligand. And then with this conjugate ready, then you can use a click chemistry reaction. You can either use biotin or this Tambra, which is a rhodamine-based dye. Click on that dye onto your conjugate and then use it further to study. So the probe that we used to study is your diazine-based probe, diazine-based probe with an alkyne handle at the end to tag on to the reporter group at the end. Again, as I told you, I am a synthetic chemist, and I would like to give you some idea of how we make these molecules step by step. So the first step is forming this photophore, which is a very highly reactive species and also highly strained species. So they are relatively unstable. And this we can get from a ketone like this. These products were surprisingly more stable than I expected, but the yields were very low, and you are left with a very little amount of product, which is not enough to study for your further studies. So the next step, as I showed you before, is a Wittig reaction. Also here where you break the double bond here to form the dialdehyde and the phosphonium salt. This reaction which as chemists would tell you, is a very straightforward and easy reaction. And I was also pretty confident when I saw it on paper that it would be pretty easy to get through, but was the reaction that I spent a considerable chunk of time, because again, there was a lot of undesired byproduct, side product decomposition. And this yield was also being so low that the next step, so two low yielding steps would really leave me with no product at all, which would not be helpful in our further studies. So, but a lot of painstaking efforts, I could get this reaction to a high 50-60% yield, which was good enough to give me a considerable amount of the final product. Now, if you see the fragments that go into the Wittig reaction, you can make the respective fragments. First is via Grignard reaction, where you add this nucleophile and then further oxidizes to get this dicarbonyl, and then this phosphonium salt, which you can synthesize from 5 steps from a commercially available starting material. Okay, so with the probe now in hand, we went ahead further to see, we were ready to find out what the protein target is. And before that, we needed a control probe for the studies for target identification. So we first chose oleomide, which is also a monounsaturated fatty acid amide. It has 18 carbon atoms as opposed to 22 in urocamide. But as you can see, the oleomide also showed similar phenotype as urocamide, but it was found that the mechanism of action of oleomide was different than that of urocamide. Then we resorted to palmitamide, which is a completely saturated fatty acid amide and which did not show any phenotype, so could work as the control. But we still continued to do our studies with oleomide, which would help us give some idea about proteins that are selective for urocamide as opposed to oleomide. And also we had another story which we were developing using oleomide. Probably I'll share that with you sometimes later. Okay, so this is the basic structure of the photoaffinity labeling studies. So as we found out from before that the microglia cells are which cells that take up uricomide. So we would want to use microglia cells, and the cell lines that we use was human microglia cell lines. The probe that we used were uricomide diazurine. The control probe was both oleamide and palmitamide based diazurine probe, and the competitive probe was uricomide itself. Okay, so all this photoaffinity labeling works very well on paper, but does it actually work in the system that we are supposed to analyze was the next question that we needed to answer. And that we did via a process of photoaffinity labeling followed by SDS phage gel electrophoresis. So first, the HMC3 cells were treated with diazurine probes. Then it was cross-linked using UV light. Then the cells were lysed and via sonification, and the proteins were harvested. And then we used click chemistry to tag on the rhodamine dyes, and then we analyzed the labeled proteins via gel electrophoresis. So as I told you, the questions that we wanted to answer is that, can we observe any labeling at all with our system? Also, what is the concentration of the probe, optimum concentration of the probes that are required to observe any form of labeling? And also, what is the incubation time of the probes with the cells that gives the desired labeling? So with that, we started our experiments, and to our delight, first time we could label some proteins. The first time we ran that experiment, we could label the proteins. The best news from this experiment was that these probes were actually soluble in ethanol, which uricomide was not. So the silicon nanoparticles were no longer needed to deliver uricomide, although it did not interfere in the labeling, but also it made our life easier. So that was some good news. And the best news was that we did observe some labeling. Now, to go further ahead, we tried to find out what is the optimum concentration of the probes that would give us the desired labeling. So we treated the HMC3 cells with a different concentration of uricomide for 24 hours. And from that, we could see that at 5 micrograms per milliliter and 10 micrograms per milliliter, we can see the desired labeling. And we chose, just to be on the safe side, we just chose uricomide 10 micrograms per milliliter as the desired concentration to see labeling of proteins. With the concentration done, the next was to find out what is the appropriate time to see the desired labeling event. So right now we treated the cell lines at different times point at 1 hour, 3 hour, sorry, half an hour, 1 hour, 3 hours, 6 hours, 16 hours, and 24, all at a concentration of 10 micrograms per milliliter. And then we examined it by gel electrophoresis and found that 1 hour and 3 hours give us the desired labeling. But again, we chose 3 hours incubation period as the optimum condition to go ahead with the further studies. Okay. So now with those conditions set, we were ready to move ahead to actually identify the target, which was done by a process called Tandem Mass Spectrometry. So this is a process where you subject your sample to a mass spectrometry process 2 times. So it consists of 2 mass analyzers which can analyze multiple samples at the same time. And what it employs is isobaric tags. Isobaric tags are tags that have the same mass numbers. So there are two types of isobaric tags that are used for tandem mass spectrometry: the ITRAC, or the isobaric tag for relative and absolute quantification, which gives us an idea of the amount of proteins in different sources in a single experiment. And the other type of tags, and the tags that we used, is a tandem mass tag, which is also a isobaric mass tag that can be used not only for protein quantification, but also for protein identification. So these TMT tags are chemically reactive stable isotopes, which mostly has three different parts. So you have the peptide reactive groups, which attach on, which tag on the peptides that we get from the proteins, which are digested and broken down into smaller peptides. Then you have this mass balance group. So for a tandem mass tag, you have a set of tags which have the same mass, and that the same mass of multiple tags is achieved by this mass balance group. And then you have a mass reporter groups which helps in the target ID of the, which helps in target identification. Okay, so just to give you an overview of how this spectrometry works. So you treat each of your sample with an individual tag after denaturizing, reducing, and alkylating your proteins. So each sample is treated with one different isobaric TMT tags, and then you combine all of them. Then when you combine all of them, and then you use that combined fraction to analyze via tandem mass spectrometry. So because all these tags are isobaric, when you pass it through mass spectrometry for the first time, it will all appear at the same time, at the same place, sorry. Then what happens, you take that peak that corresponds to these tags, and then you break them up again and then analyze it again with mass spectrometry. So now it breaks up to the corresponding fragments, and each corresponding fragment has a known reporter ion, which is specific to tags that were treated with individual TMT tags. So, and if you compare the ion peaks that you get from these reporter ion peaks, and then compare it to the databases of peptides already present, you can identify the proteins that has been tagged in each sample. So this is the process that we use for the target ID. Okay, again, to run you through what happens is you first treat your HMC3 cells with diazine probes at the concentration that we found out optimal was 10 micrograms per ml. Then you incubate it for 3 hours. Then again, you cross-link it. You break the cells down. Then you normalize all the protein concentration to 1 mg per ml. And then you use click chemistry to attach biotin. So previously we were attaching fluorophore dyes. Now we are attaching biotin and then enriching it using streptavidine beads. And next, we break down these proteins into the corresponding peptides. Then we attach the TMT tags, and then this sample is run sample is put through mass spectrometry to find the proteins that are tagged in this experiment. Okay, so with our experiment, a total of 4,735 proteins were pulled down. But these were pulled down by all the 3 probes that has been employed in the studies. But then we needed to narrow down the proteins of our interest, which would be specific for uricamide as opposed to the other 2 probes used. So with that data, we imposed some restriction conditions, like the intensity of uricamide and palmitamide greater than 2 and uricamide and oleamide greater than 1.5 to pick out the proteins that are selective, and the total number of proteins from that list that we got was 65. And we analyzed all these 65 proteins that we got, and we tried to find out which category of proteins which category these proteins belong to. We were happy to see that there were some transporters and membrane trafficking proteins. That is what we would expect uricamide's target to be. And then we also analyzed the various functions that the proteins were involved in. And you had quite a few proteins were involved in signaling and cellular processes, signal transduction, transport. So with this results, we further shortlisted, imposing more restriction to a group of 11 proteins. And I don't think I can talk to you more about the proteins because it is something that is still ongoing. And we are still, we want to zero down and confirm before we share this result of what protein has been isolated. So I will just give you a gist of what is going on right now, how to actually validate the protein that we validate the protein interactions with uricamide. The first step, what we did was to find out that amongst the top 11 candidates that we have, were they expressed in retina at all? But to our dismay, that all 11 were expressed in retina, and that was practically a dead end. But then we started to see if these 11 candidates can activate microglial cells. And we were very happy to see that one of the proteins of the 11 candidates was activation of microglial cells. And it was found it was important for microglial cell, activation of microglial cells. So right now, we are studying this protein in depth. We are trying to see the binding interaction with uricamide. Other experiments are is undergoing right now to see whether this protein can be the protein that is of our interest. Another way of validating all these 11 candidates would be to use photoaffinity labeling studies with retinal tissues. So all the photoaffinity labeling studies that have been reported, that have been done, it's mostly done on cellular system. Nothing has ever been done in tissues or in tissues. So if we can get the studies done with retinal tissue, which will be the first of its kind, we have been successful in labeling proteins when we do retinal tissue photoaffinity labeling with retinal tissues and with gel electrophoresis. Right now, we are doing the photoaffinity labeling with tandem mass spectrometry. So after that, we would like to compare the list of proteins that we get from the mass spectrometry studies from the tissue and the cell lines and see whether there is overlap at all between these two protein data that we collect. And that would also help us validate and narrow down proteins of interest for uricamide. And another aspect of this project that is still underway, it's mostly chemistry, so I'll just give you a brief gist of it. So we also want to see if we can make better and more efficacious analogs of uricamide. So these bioactive lipids or bioactive amides are degraded by an enzyme called fatty acid amide hydrolase. These enzymes, they hydrolyze the amide bonds forming the corresponding acid, and they are degraded in the system. So a major way to improve the metabolic stability of all these amides, especially these bioactive amides, is to make the amide group less labile to hydrolysis. And a way of doing it is introduction of oxygen in place of carbon here to get what we call the carbamide derivative, and introduction of nitrogen in place of the oxygen, which is called the urea derivative. So we have this molecule in hand, and this molecule, the carbamide derivative, has preliminary results have shown that this carbamide derivative is more efficacious than uricamide. So down the road, our aim is to make analogs of uricamide, which are more stable towards SPA, and also to make analogs that can enhance the binding to the target protein by rigidifying the structure somewhat. And the most important part that we want to address is increasing the solubility of uricamide, which is a major hindrance in studying this lipid. And if we can incorporate this, all these features and make an analog that can be used for down the road in treatment of retinal degeneration. With that, I would like to thank my postdoctoral advisor, Professor Dale Boger, who's been extremely kind, extremely supportive. Our lab is mostly a synthetic chemist lab, but he's given me the total freedom to do research, whatever research I like, helped me move into these different fields and given me whatever support I needed. And our lab manager, Ezra Salzar, she is on top of her game with all administrative stuff, and she makes our life way more easier. All the members of the Boger lab group, past and present, they are an extremely talented group of synthetic chemists. They're very supportive, very motivated, and it's been an absolute pleasure working with all of them. I would also like to thank our collaborator, Professor Martin Freelander. He has not only provided us with all his expertise, knowledge, but also all the funds. As you would know, these research are not expensive. Everything that you use are very, very expensive, but he's really had no objection and really been very helpful. And the postdoctor in Professor Freelander lab, Dr. Guochin Wei. It's an absolute delight to work with her. She's entertained all my queries of, a novice synthetic organic chemist trying to be a novice biologist. She's been very patient with all my questions and making me understand and walking me through all the biological processes that's going on. Professor Benjamin Cravat, without his guidance and prized opinions, we would not have been able to take the project to the direction that we are at. And also the postdoc in Dr. Kravat's lab, Daisuke, and again, very helpful. He's helped me with all the biochemical techniques that I've shared. He's helped me learn everything, very patiently taught me everything. And last but not the least, I would thank you all for hearing me out, giving me this opportunity to share my research with you guys, although not being from a biology background, but helping me show that chemists are also, chemists form an integral part of the biology community as well. And Arup sir for giving me and inviting me to give me the lecture, and everybody here, all the staff members and the students, the organizers of the event, thank you so much for the opportunity, and I'd be happy to take any questions if there's any.
Thanks a lot, Shrosri, for your very wonderful presentation. There you have started from scratch. Really, it was nice to learn the basis of retrosynthesis, right? And also you have started with how to carry on the labeling. You have started from scratch, and later on you have finally said that how 11 proteins on which you are working on, and they can be used to actually understand the role of uricamide in retinal homeostasis, and what else can we do. You have generated this, say for example, derivatives, carbamide derivatives and urea derivatives, and you are finding that they are giving you very promising results. So really it's high time when chemists and biologists should join hands together, and definitely then science will proceed in a much better way, right? So we are really fortunate to have you, and it is also we are really thankful to Orubda that we are getting the opportunity to interact with those students who were having chemistry as their main paper and microbiology maybe as their past paper, but definitely that little contribution has left so deep impression in the minds of those students that even after so many years, whenever they get a call from Aurup sir or from St. Xavier's College, in spite of having so busy schedules, you every time we get you people interacting with our young minds and inspiring them. So we eagerly wait for the day when we will be in a position to meet with such students in an offline mode. And thank you from my end and from my all the faculty members of St. Xavier's College. And it was really a wonderful listen from you, starting from chemistry and how it is actually helping us in understanding different curing methodologies for different retinal disorders. Thank you, Chemistry.
Thank you.
I would request MMG ma'am to kindly felicitate Dr. Sreyoshri Chatterjee.
Thank you, Dr. Sreyoshri Chatterjee. It's for you.
Thank you so much. Thank you for inviting me, and thank you for giving me this opportunity. I'm really honored. Thank you. And thank you, Aurup sir, for giving me this opportunity to present this research, even though I'm not from a microbiology background.
Thank you. Thank you. It was great listening to you. Right.
I would like to thank Dr. Sreyoshri Chatterjee for delivering such an informative and interesting presentation to our audience, and our moderator, Dr. Mahashweta Mitra Ghosh ma'am, for wonderfully conducting our second technical session. Now, we would proceed on with the third session of Practice in Biological Sciences, Chapter 5. I would like to call upon our moderator for this session, Professor Dr. Madhumita Moitra ma'am, to introduce our last speaker for the day, Dr. Asif Ali, a postdoctoral scholar from the University of Chicago, Illinois. He would deliver a presentation on the topic, Sticking Together in Subcellular Space to Survive Stress, an inbuilt biomolecular phase separation program as cell survival strategy. Over to you, ma'am.
Thank you, Srinjaka. Thank you. Do we have Asif here?
Ma'am, Dr. Asif Ali is
Srinjaka, you have to unmute yourself. Yeah.
Yes, ma'am. Ma'am, Dr. Asif Ali is facing some technical issues. He will join us shortly.
Okay.
I would request the audience to wait for some time. Dr. Asif Ali will be joining us shortly. Meanwhile, I would request our audience to kindly like and subscribe the official channel for the Department of Microbiology, and stay tuned. We have Ma'am with us, and Dr. Asif Ali will be joining us shortly.
Srinjika, you are there? Hello.
Yes, sir, I am here.
Dr. Asif Ali didn't join?
No, sir, actually, he is facing some technical issues. The organizing team is trying to contact him. So he said that he will be joining shortly, but it will take him some time, a few minutes maybe.
Okay, okay. Thank you. Thank you.
Okay, sir.
Hi.
Okay, Asif.
Oh, yes, ma'am. Sorry, I just finished my talk in Boston Station.
Okay, okay. Okay, good to see you, Asif. Good evening.
Good
evening, everyone. Good evening. So it's a very proud moment for me to introduce Asif today as Dr. Asif Ali. Good. So let me introduce Asif as our student. After completing his bachelor's in microbiology from Surendranath College, University of Calcutta, he was our student in master's. He was the first batch of the master degree students in our department in St. Xavier's College. And after that, he completed his PhD in Bose Institute under the supervision of Professor Mahadev Pal. And in his PhD work, he did a very, very good thing. He was working with the proteotoxic stress response, and he found that the heat shock factor 1, the master regulator of PSR, induces NF-kappa beta signaling by directly inducing the transcription of tumor necrosis factor gene in response to the proteotoxic stress. And after that, he continued working in his academic career. He has got a very good academic career. He is now working as a postdoctoral scholar in the University of Chicago in molecular genetics and cell biology division in the lab of David Pincus. And he's still doing a very, very good research. And today he is among us. So I really feel very proud to see Asif today in front of us. And we are eagerly waiting to learn the research that he is doing. And he will be talking on the topic that is sticking together in subcellular space to survive stress, an inbuilt molecular biomolecular phase separation program as a cellular survival strategy. So Asif, over to you. Let us hear the research, and we are eagerly waiting to hear you. Over to you, Asif.
Yeah, thank you, ma'am. Am I audible to everyone?
Yes, yes.
Yeah,
yes,
awesome.
We can hear you.
Awesome. Let me start sharing my screen. So you people can see the slideshow, right?
Yes, yes.
Yes, sir.
Awesome, great. Yeah, first of all, I want to thank St. Xavier's College Biotechnology and Microbiology Department for giving me this opportunity. It's always a pleasure to homecoming and delivering what you have studied in past 7 to 8 years. So what we are going to learn today is sticking together to survive stress. Although this topic is not complied with the current situation where we don't have to stick together, we have to maintain social distancing to fight with the pandemic. But believe me, I'm going to show you that sticking together can help us in some stress conditions, and also it helps cells and biomolecules to really help each other. So if you consider cell as institute, and if there is any kind of insults happen, if we stick together, we will have a very good growth report. But if we don't, then we are gonna not survive the stress. Let me first put the problem in front of you. A single string of amino acid of the protein must properly fold in its 3-dimensional to be functional and to be used by a cell. But this folding has lots of intrinsic challenges. One of the challenge they poses is highly crowded cellular environment. What you're looking over here is a picture of a cytosol taken up, and you can see that how crowded this cytosol is with different kinds of protein. In average, cytoplasm of yeast contains 250 milligrams of protein per ml, and the volume it's supposed to fit in is 40 cubic meter. So we can understand how much crowded the environment is. It's not only about crowding, it's also about highly dynamic and highly kinetic environment, because each of them are working together and doing some other work. This movie, which you are looking, it just slowed down for our visibility, but actually the real scenario is 1 million times higher of this speed. So we have to multiply the speed 1 million times to get the real cellular condition. So this whole condition of highly crowded, dynamic and kinetic environment is very prone for aberrant interaction and toxic aggregate formation. How cell really keeps the cytoplasmic or keeps the protein soluble. There's a designated protein system called molecular chaperones, right? These are the proteins that helps protein to fold. So protein helping protein to fold, and those are molecular chaperones. So we have HSP 70, HSP 40, HSP 19 chaperonin family of proteins in the system, in the eukaryotic system, that really helps the cell to keep a soluble form of all of these proteins. Right? Let's deal the problem at the molecular level. There's another problem beyond this crowding and high kinetic environment, is that the translation itself is a slow process. It takes 10 seconds. So for a multi-domain or high structure proteins, the folding should occur co-translationally. Right? If it is happening co-translationally, it is potentiated by a group of chaperones called RAC and NAC. RAC is ribosome associated chaperones and NAC is nascent chain associated chaperones. So instead of getting its final working conformation, they got a folding intermediate, and they should get its native conformation with the help of HSP 70, and the J proteins really help 70 to keep it. And then sometimes it also requires the help of HSP 90 chaperone and chaperonin system to reach the native conformation. So this is the whole cycle, how really co-translational folding in that highly crowded environment is taking place to really protein get this native conformation. However, this folding intermediate is also very prone for misfolding. And if it happens, it can go for an aggregated conformations. And then again, it's HSP 70, J protein, and HSP 104 helps this aggregated conformation to get back to folding intermediate and push this equilibrium towards the native conformation. But sometimes the aggregate is overwhelmed and we cannot handle it, right? So these systems, the HSP 70 and J protein, along with an HSP 104, which is a disaggregase, just untangle this aggregate and can help them to be degraded by proteasome or by autophagic degradations. So this is all about talking about protein homeostasis, how the proteins are properly folded and in the crowded environment. So in a healthy cellular condition, the protein folding demand and the protein folding capacity are in fine balance, right? The capacity is by the available chaperones in the system. However, during stress, there's a lots of accumulation of non-native protein conformation, and the demand goes beyond capacity. Mostly we understand this thing with the help of heat shock by increasing the temperature from being at ambient temperature to any kind of insulative temperature, and the cell feels sick, right? Under these conditions, there's an evolutionarily conserved transcription program which is governed by a transcription factor called heat shock factor 1, which is a winged helix loop helix transcription factor that binds to its congruent DNA element called heat shock element and induces the transcription of chaperones in the system. And then that helps to bring this whole homeostasis back to normal. Once the homeostasis is restored, the chaperones comes and do a feedback inhibition of heat shock factor 1. So the two questions we are going to understand today: what really keep it repressed after the stress, and what really signals heat shock factor 1 activation. Why these two questions are important? Why we need to study heat shock factor 1 regulation at first? Let me tell you that heat shock factor 1 activity is directly linked to two diverse kind of disease, either cancers or neurodegenerative disease. These two diseases are phenotypically diverse. In one condition, there's a hypergrowth of cells. In another condition, it is happening in the aged cells or the slow proliferating cells. In one condition, HSO1 is hyperactive, and in another condition, HSO1 is inactive towards these amyloid aggregates. These two diseases can be linked by one molecule, that is HSF1. So if we find a drug, or if we find a solution for this HSO1 regulation, we can find a one drug solution for a wide kind of a disease. So we don't directly use these models at first. We used yeast as our superhero to let us understand how HSO1 works in their system. And once we found that, we imply those knowledges on this disease condition and try to help this up and to try to have a kind of a solution for this disease. So the first question we have, what regulates Hitch factor 1 activity? The first and straightforward approach any molecular biologist will take is that perform an immunoprecipitation of the factor, right, which we are interested in. So we are interested in Hitch factor 1. We tagged it with the 3x flag at its endogenous level with the help of CRISPR, and we either left the cell untreated or give a post-stress, means we give heat shock and wait it for 1 hour. Anything which will interact over here, we can affinity capture and then perform a mass spec to identify what those factors are. So you can see that in one axis, we have stoichiometry of HSO1 interactors at 30 degrees centigrade and at post-stress, right? Control and post-stress. And what we found above this cloud of noise, the most abundant interactor of HSF1 is HSA1, 2, and 3, and 4. Right? These are actually the family of HSP70, which I just introduced you. So if these are really the repressor or inhibitor of HSF1, if we delete these genes one by one, we should get the activity of this under non-stress condition, right? So what we did, we deleted all these one by one, and you can see that the most abundant protein among this paralog of HSP70 is SSA2. Deletion of this gives the induction of a reporter of HSF1. So as I mentioned you before my introduction, that HSF1 binds to a DNA element called heat shock element. And if we put an YFP reporter in front of this, we can measure its activity. And we found SSA2, the 99% of the protein of this paralog is then SSA2, and that gives the induction of 4-fold. And if we code SSA1 and 2, the constitutively expressed gene, we get a higher and synergistic effect. So this biochemical evidence that is HSA1 is getting repressed by SSA1, 2, and 3, and 4s, and also this genetic assay confirmed this finding. So these two observations made the field of this chaperone biology or HSF1 field to draw a model, which is called HSP70 titration model, HSF1 regulation, where under non-stress condition, 70 keep HSF1 repressed and there's no heat shock response. When there is a heat shock, any kind of disturbance to the environment, 70 clients accumulates and 70 gets stated away from HSF1 and HSF1 get activated. In turn, HSF1 induce more 70 level in the cell and 70 keep it repressed further on. And this is called feedback loop, right? So when I joined David lab, this was one of the most preceding and one of the most preliminary model people have. But there's a lots of challenges this model, and there's lots of conceptual challenges this model has. One of the model which I found is that the number of HSP70 and HSF1 is in a ratio of 1000:1. And HSF1 binds with HSP70 in macromolar affinity. So when we give any stress, it's supposed to titrate away 99.9% of 70 away from HSF1. Second, HSF1 resides in the nucleus. 70 is partitions throughout the cell. And it has been believed till that time that cytosolic protein titrates 70. HSF1 is activated within 2 minutes. So what we asked from this model is that within 2 minutes, 99.9% of 70 should move away from HSF1, which is conceptually challenging. And whenever we try to have this model and perform a mathematical model with this, we always fail to have a mathematical model working. So how can so much 70 be depleted from the nucleus so fast? Let's understand how 70 works. Any substrate of 70 can either bind to 70 in ATP or ADP bond state. ATP bond state, the affinity is kind of low, but in ADP bond state, the affinity is 10 times higher. Right? Its KD of efficiency is 1 micromolar. J-domain proteins is actually an ATPase that basically helps 70 to degrade this ATP and couple this fast K on with the slow K off to get a 1000-fold higher magnitude of affinity, and that's around 10 nanomolar. So usually for the 70 working model, there's always a J-domain protein that helps them to keep this cycle and provides an ultra affinity to this model. What the field and what the scientific community is missing that time is a J-domain protein role in this system. So we look for that. We want to find what are those J-domain proteins. So to understand the role of J-domain proteins, we took an approach called anchor away approach, right? Where we tagged a series of proteins and chaperones with FRB, and another domain called FKBP we tagged to the ribosomes itself. Right? So if we add rapamycin, we all know that an addition of rapamycin will heterodimerize FRBP with FKBP, and as quick as within a minute, as ribosome will export out to the cytosol, this protein will get out from the nucleus to cytosol. This approach is called anchoring away any kind of protein from the nucleus to cytosol, right? So what will happen if I add rapamycin? So as I told you, HSO1 is totally nuclear. So any factor which is nuclear will be the regulating HSO1 activity. So if I add rapamycin, what we will see is that this protein get anchored away with the ribosome from the nucleus to the cytosol. So we just tried this approach with all the known chaperones in the system. We tried with HSP70, 90, and other systems. What I want to show over here is that we tag SSA with FRBP, where we have ribosomal protein with FKBP, and you can see that this magenta color is SSA, and you can see it's throughout the cell, right? It's only not in the vacuole, but it's everywhere. HSF1 is mostly in the nucleus. You can see the green over here. Now, if in normal condition, this green perfectly co-localized with the magenta, and we can see the green co-localizing with magenta will become white in color. When we add rapamycin, we are anchoring away this nuclear to cytosol, and now the white becomes green. So it means that there's no chaperones on top of SSF1. And we tried this with tons of different proteins. One of the J protein which is most abundantly nuclear is cis1. Cis1 is an HSP40 proteins in the yeast, and we found that we can anchor away this also very efficiently. Right? We can see it's in the nucleus. When we add rapamycin, we can take it. We can see the hollow nucleus, and it becomes totally green when we have rapamycin in this. So under this condition, we just went to our reporter system. We have HSA1 reporter system. So now we are anchoring away HSP90. There's no effect. There's no induction of the reporter. Now, if you anchor away cis1, there's an immediate and a sustained activation of the reporter. And this suggests that nuclear residing cis1 is really inhibiting HSA1 activity. But now this is a reporter system. What happened globally, right? So we performed an RNA sequencing of whole RNA transcriptome of the yeast under a cis1 non-anchor away or anchor away condition. If you follow this axis, anything in this axis is unperturbed. And we can see most of the genes are unperturbed if we add rapamycin. However, there's an upregulation of HSA1. All these red dots are HSA1 targets. So with this RNA seq analysis, we can confirm that HSA1 induction is very much specific by cis1 anchoring away from the nucleus to cytosol. Remember, we are not giving any stress. We are just making this protein out of the nucleus, and that is sufficient for activation of HSA1. All good. We just found a model that say that, okay, nuclear residing cis1 keep and potentiate 70 repressive effect of HSF1, right? If that's true, we always, when we pull down HSF1, we find SSA bind, right? But if you anchor away cis1, the binding of HSA1 with SSA decreases. It's perfectly comply with this model that cis1 is really what is potentiating this effect. It's not about 70 binding directly to it. That's good. So now we have a synthetic model. We can call it as a chemical synthetic model, where if you synthetically remove cis1 from nucleus to cytosol, that is sufficient for HSO1 activation. No need to do any stress. We can just activate it. But is this physiologically relevant? Is this really cis1 relocalizes to cytosol during stress? To understand that, I tagged cis1 with YFP, and yellow fluorescence protein, which is shown as a gray color over here. and HSP104, which I told you as a disaggregate, so it will work as an aggregate marker in the cell. So now what you're looking in this cell is cis1 is totally in the nucleoplasm and very diffuse signal in cytosol. Okay, so what we are expecting over here, when I will give heat shock, this protein should move to cytosol, right? Because when we anchor away this to cytosol, we can activate heat shock factor 1. We know when we give heat shock, HSP1 get activated. So this protein should move. So let's watch together what happens, right? We give heat shock and we keep giving, and oh no, it's instead of going to cytosol, it is actually getting relocalized to a subnuclear region. It's moving away from here for sure, but instead of going to cytosol, it's really forming a subnuclear ring. And this was very surprising result for us because we are expecting, because as I told you, the model is that cytosolic disturbance is attracting chaperones towards it, and that's what HSP1 get activated. Okay, so let's hold our breath for a while. Let's first confirm that if it is moving away, it is sufficient to activate HSP1 or not, right? It is moving away from HSP1 or not. So we tagged HSP1 with YFP, with cis1 being MS Scarlet. You can see red over here. So in non-stress condition, at a normal condition, when there's no heat shock, this and green overlaps. So if you draw a line over here, you can see an overlapping signal of cis1 with HSP1, right? So it suggests that cis1 is keeping it HSP1 repressed. Now, when we give heat shock, we know that it forms this ring, but it's moving in subnuclear region, but still it's moving away from HSP1. As when we draw a line over here, we can see that the signal of cis1 is really partially away from each other. So that's good. Our model is okay, that cis1 is moving away from HSP1, but instead of moving to cytosol, it's moving to a subnuclear region. So we can just redraw the model. So now we have this protein in the model that suggests that cis1 actually potentiates 70 effect on HSP1 and keep it repressed. During heat shock or any environmental condition which is disturbing for the cell, it's not about 70 clients, it's about cis1 clients. And now this model perfect fits because cis1 is nuclear, right? And it's totally nuclear, it's mostly nuclear. Cis1 HSP1 ratio is 10:1, and we found that the relocalization is within 2 minutes, so it's very fast. So only thing we need is subnuclear relocalization for HSP1 activation. If this is true, can we now make a mathematical model with this thing? Yes, we can. So this is the mathematical model we have in the wild type condition. When you give heat shock, there's an induction of HSP1 reporter, and that gets saturated over time. Now, if you have a nuclear localized cis1, now we tag cis1 with NLS, right, which will keep cis1 always in nucleus, will not allow it to go to any other place. We don't get a maxima output of the HSP1. If we have NES cis1, what we saw is that it's getting much more upregulated. So changing the localization of cis1 from nucleus to cytosol, we can modulate HSP1 activity. And this is all what I'm showing you is a mathematical modeling simulation we have run. And if we perform the experiment, we perfectly mimics this model. That in the wild type, this is the curve. When we have NLS, it's really less. And when we have NES, it's perfect, much more upregulated and much more induced. So that's good that this model is perfect and we can specifically solve the conceptual challenges we have. Now, that's not the first thing which we are searching for. We're searching for what is sticking together, right? So, but first of all, we have to correct this model. We are not in a good, and we're all on the same page that we have a corrected model of heat shock response. What are these clients? Where these are moving? It's not moving to cytosol. It's moving to a subnuclear regions. But why and where? To understand that, we performed a in vivo proximity labeling to find the cis1 interactome. Okay, so classical pull-down experiments have an artifact that is called cell lysis artifact. What this artifact is? Let's say a protein A resides in nucleus and protein B resides in mitochondria. When we do pull-down, what we do? We lyse the cell. So now mitochondria and nucleus is lysed. Now they can come together, and in our pull-down experiment, we can find A and B interacting. But actually, they are in 2 different compartments. That's not true. That is called cell lysis artifact, where we have a kind of inside the lysate they are interacting, but inside the cell they may not interact. So we perform this in vivo labeling. How we can do it? We tagged cis1 with an enzyme called Apex2. Apex2 is a peroxidase. So if we add H2O2 to the system and we add biotin phenol, any protein which will come to the vicinity of this will get biotinylated. And we can pull down these biotinylated stuffs and we perform the mass spec. So this is different from the classical pull down where the labeling is happening in live cells, not in the cell lysate itself. With this approach, we're able to find lots of interactome of cis1. Anything which is, so this is what you are seeing is a volcano plot. So anything toward the positive axis is the interactome of cis1. We found ribosomal proteins, we found lots of nuclear factors, we found chaperones, we found translation factors, we found ubiquitin proteasome systems. Let me just categorize all of this for you for better understanding. We found the components for chaperones, found the components for proteasomes, which really helps to degrade the stuffs. We form transcription factor like PAF complex. We form nucleolus factors. We form ribosomal proteins also. And when I tag protein from each of this family, that I tagged the CDC48, LTN1, RTN1, RPN1, and when we perform heat shock, we found that it's a perfect cytosolic foci formations with all of this co-localized with each other. At the ring structure, our subnuclear region, we found that proteasome is a part of the subnuclear regions. So that's good that our proteomics and cell biology is very perfectly matching with each other. And if I perform and show you a model of this, that what we're looking over here is in ER, where I mark with RT1M scarlet. So cis1 this is the three-dimensional relocalization of cis1 during stress, and this is a three-dimensional relocalization of HSP104. And if we merge together, this is what it looks like. And if I slice this down, you can see that there's a subnuclear cis1 ring and cytosolic foci, and all of this happens on the surface of ER. So we have a contiguous network from nucleus to ER of cis1. Right? So that's good that cis1 relocalizes away from HSO1. That's important for cis1 activation. So now we can have a very small model that suggests that under non-stress condition, cis1 remain in the nucleus and potentiate 70 repressive effect on HSF1. When we give heat shock, there is an relocalization of cis1 from nucleoplasm to perinuclear regions. And in doing so, 70 effect on HSO1 is released. The next question we are raising up is, what are those factors here that really attract cis1? So again, we took the approach to, it is called micrographic approach to understand what are those new steps where cis1 is going for. So we tagged cis1 in our tetracolor strain with the green. HSP104 is over here blue, and in the white you can see NSR1 and Sex61 is red. So in non-stress condition, cis1, we can see this is the nucleoplasm and this is the nucleolus. So nucleolus in the east looks like a crescent shape or sickle shape structure. Non-stress condition, cis1 is concentrated in the nucleoplasm, very low signal in cytosols. When we give heat shock, as quick as 2 minutes of 39 degrees shift of this is sufficient for this perinuclear and subnuclear reorganizations. You can see that it's moving away from nucleoplasm to subnuclear regions. If we keep continuing the heat shock, it forms cytosolic foci. So this cytosolic information, which people are thinking before and which we had the model system, is happening later on. Now, if I told you before that this folding problem is co-translational. So I treated the cell with cyclohexamide. Cyclohexamide is a drug which blocks the synthesis of the protein. And if I treat it 5 minutes before giving heat shock, what I see that the cis1 really don't move to any other structure. Neither it forms foci, nor it forms these structures at all. So even though temperature is there, cis1 don't move at all. So this is something which suggests that it's really a step which requires an active translation, right? It is prerequisite. So if the active translation is really what is we required for this happening, we performed a cis1 polarity index analysis. So we can see that cis1 is mostly nucleoplasm, now in the subnuclear. So if we go for cis1 polarity, we can see that it's non-polarized during non-stress condition and get polarized towards subnuclear region. And the polarization sustained for 10 minutes of heat shock. But if you add cyclohexamide, there's no further polarization at all. So what we learned from this experiment is that cis1 polarization requires active translation. So if you remember, I showed that in my pulled on Apex2 experiment that we got all the components of ribosomal proteins, right? Yeast has 71 different ribosomal proteins, and it forms a 1 megadalton large complex of ribosomes. Each of these components are found in my cis1 pulled on. So we get tempted to see what are the status of this in our imaging strains. First of all, we perform the mass spectrometry again by treating with cyclohexamide in a cis1 flat tag construct. So we are looking at the stable interactome, and we are having heat shock or cyclohexamide pre-treated and then heat shock conditions. So again, we are looking at a volcano plot. Anything towards this negative axis are translation independent. Anything in this positive axis are translation dependent. And what we can understand is that these all pink circles are ribosomal proteins. So all the ribosomal proteins interactome requires active translation. So now we just went through to tag ribosomal protein with a tag called halotag. Halotag is actually an halogenase, which is an enzyme. When it forms any chloroalkane in vicinity, it forms a chemical adduct. So this protein itself is non-fluorescent. We cannot visualize in the microscope. But if we add a fluorescence dye like JF549 with this, we can perfectly visualize it. Right? So now what we are looking over here is already heat shocked cells, right? Already stressed cells. So we can see a subnuclear ring. We can find a cytosolic foci. But this ring and foci is not at all co-localizing with the ribosomal proteins. So it's again very surprising for us that it's not true what we thought before. We got our clue from our cyclohexamide experiment that it's an active translation that is required for this cis1 polarity. So if that's true, then maybe it's a newly synthesized ribosomal proteins that should deposit over here. So what is happening over here is that cell has 99% newly mature protein and only 1% is newly synthesized. So it's kind of a darkness below the line. If there's tons of this, that this chloroalkane is very saturated for the old one, we cannot see the newly synthesized. So what we did, we add a blocker. The blocker, we add this. Now we cannot visualize the mature one, right? They are there, but we just block them up. Now when we add the new dye, we can see only newly synthesized proteins, and we can see now that there's no co-localized with the mature one, but newly synthesized perfectly co-localized with the cis1. And this thing is not only true with the RPL26, also true with RPL25, RPS2, RPS4B, but this is not true with the ribosomal protein like RPL29 and RPS3. These two are the one which takes part in the late ribosome assembly. So what I'm showing over here to you is that during the heat shock, newly synthesized ribosomal protein get co-localized at this perinuclear region with cis1. What's the fate of these proteins are? What will happen to them if I continue the stress, right? So we first of all performed a simple biochemistry experiment to understand that really these proteins are the true interactor of cis1. So we labeled either old or new protein and perform the immunoprecipitation. So you can see that compared to input, we have enrichment of cis1. But when we have old ribosomal proteins tagged, we don't see any immunoprecipitation with cis1 pulled on. However, if I have newly synthesized protein tagged, we can see them getting enriched. As quick as 2.5 minutes of stress is sufficient. So now we got the answer that the client for cis1 is newly synthesized ribosomal proteins. So now the question is, what is the fate of this? We performed a simple experiment called pulse chase experiment. which is a kind of different from the classical pulse wave experiment where people add cyclohexamide for other kind of stuffs. But here what we are doing is that we have ribosomal protein tagged, we add the blocker, wash it out, and add the new dye. So now we can pulse for the newly synthesized one. Now if I add the blocker and chase down during the heat shock course of period of 10 minutes, right? Let me first tell you that in yeast, every minute there's 2000 ribosomes are made. So ribosomal proteins translated in the cytosol, get imported in the nucleolus, get incorporated on the ribosome, and then get it exported out. This whole process happens every minute for 2000 ribosomes. And so you can say that each subunits get incorporated within a milliseconds. Now, if I have a newly synthesized ribosomal protein labeled and performing a chase experiment, what we are seeing over here is that the blue over here is NSR1, the red over here is NSRP, and cis1 is over tagged over here. So the NSRP is neither getting inside the nucleolus nor getting exported out to the cytosol. So it's looking like they are preserved or getting stuck at this perinuclear structure, and cis1 is always doing a kind of a molecular stir bar event on that to keep it soluble all the time. So this is pretty much kind of a surprising result for us, because it's really the entry of ribosomal protein inside the nucleolus is getting denied at the time of heat shock. So we perform the same pulse rate experiment. Here we don't keep the cell in stress, we keep the cell in the recovery. So if we have it in a stress condition, we see a perinuclear deposition, but if we do a recovery, it gets exported out to the cytosol. So we can say that this preservation is reversible, and it can perfectly get exported out to the cytosol when the recovery is happening. So if this is true, and this is one of the mechanism of HSO1 activation, let's have a cell which will produce less ribosomes. If cell will produce less ribosome, then there should be less induction of HSO1, even though we are giving heat shock. So the sole transcription factor which is responsible for ribosomal protein genes transcription is called IFH1. So we tagged IFH1 with a tag called auxin inducible degron tag. Now, if you add an ubiquitin ligase called OST1 and an auxin called indole acetic acids auxins, in the presence of this auxin inducible degron will degrade IFH1 with the help of proteasomal degradation. This degradation will happen as quick in 5 minutes, right? So compared to other classical genetic models or a way of genetic deletions, this is much more accurate and much more fast and much more acute. Why we need this approach? Because ribosomal proteins are essential for cell. We cannot delete this protein one by one. So we just deleted this for 5 minutes and try to understand what is happening. So no IFH1, less RP production. And if you perform an RPB1 chip and we perform an RNA experiment, we can see that ribosomal protein expression is low. Everything else is not getting perturbed. So now we just tested our cis1 polarity under this condition. So we know in non-stress condition, cis1 is in the nucleoplasm away from nucleus. When you give heat shock, there's a perinuclear deposition of cis1 surrounding the nucleus, the subnuclear relocalization. But if you add auxin to deplete ribosomal protein production, and even though you are giving heat shock, any kind of stress, cis1 is not getting polarized. So we are basically changing the central dogma of stress response, that it is the temperature or heavy metals, any kind of, it's not about that. It's not about the physical values, it's about the biological values which is accumulating over here. So temperature doesn't matter. What matters is accumulation of newly synthesized ribosomal proteins. If that's really true, then if I measure the HSO1 activation by looking at SSA4 qPCR, we know that when we give heat shock, this gene get induced as much as 1,100 folds. Now, if you add cyclohexamide, there's no nucleosynthesize, there's a 50% reduction. Now, if you deplete IFH1, if the cell produces less ribosomes, the whole response is 50% less. So we can say that the cis1 relocalization contributes for 50% of heat shock dependent HSO1 activation. So the next thing come to our mind is that, okay, all the time ribosomes are getting synthesized, getting assembled. what is new about during heat shock condition that attracts or titrates cis1 towards it. So we performed an experiment called in vitro liquid-liquid phase suppression condensate analysis, or a kind of in vitro condensate analysis, where we had the same approach of labeling either old or new ribosomes. Now this time we'll cryolyze the cell. We just crack open the cell, dissolve in a physiological buffer. Now instead of heat shocking the cell, we are heat shocking the lysate. And let's just go and see in the microscope. What we are seeing over here, the mature protein looks like diffuse and nothing. But the newly synthesized one form these clusters. And this is called wetting of surface. And wetting of surface means that they are liquid, right? But if you see, if these are liquid in for sure, then they should come together, right? Because two liquid droplets at any time will come together and will form a one droplet, right? And this event is called fusion. So if you watch this movie over here, over a period of time, these two condensates come together and form one condensates. So it means that all of these are liquid. And if they are liquid, they all will form fusion. Like you can see over here, they form a necking over here, and then now these two really separates apart. So they're basically intermixing and they're dynamic. So they are really going for fusion and fusion events. So we can say that liquid-liquid condensates. So what I showed you on those subnuclear structure, they are actually liquid-liquid phase separated compartments. And this is a new kind of organelle, which is membraneless, but it still have some attraction towards some protein and exclude some other kind of proteins. So why I'm saying liquid, right? Can we convert it to solid if there's no chaperone in the system? So we just perform the same in vitro experiment. You can see there are droplets, right? If I deplete ATP, we know chaperones require ATP for its function. We added an enzyme in the lysate called apirase. Apirase will chew up all the ATP from the system. When the ATP is depleted, this liquid thing now looks like an amorphous aggregate and looking like a solid flakes, right? Not only by ATP depletion, if we inhibit HSP70 or we inhibit the co-chaperone cis1, all of this will convert this droplet-like structure to like an amorphous aggregate, but not if you have HSP90 inhibition. So this liquid-liquid phase separation is unique for 70 and HSP40 system. So now I'm just saying to you that this is liquid, this is solid, by just looking at the images, right? So that's not one of the best way to show. So when we took that approach, we performed a movie of this condensate. I can see that this, in a not treated condition, we can see they are intermixing, they're highly dynamic, they are really moving. So we can call them liquid because solid cannot go for intermixing or mixing within each other. But you can see the solid, they're hardly anything. All of this we are seeing is totally a noise. So if I perform a pixel by pixel variation analysis, the more brighter the color is, the more variable they are. So liquid condensates are really dynamic, but the solid ones are not. So we can say that HSP70 chaperone system is working as a molecular stir bar. It is stirring all the time and keeping the NSRP in the liquid form. But if you deplete this, they just change the shape and they become solidified. So if this from liquid to solid condition happens, can cell use it up? Can this is still usable or not? So we performed the experiment inside the cell where we have heat shock cell, cis1 is green over here, NSRP is red, and we have nucleus as a blue. And you can see that there's an accumulation of or perinuclear accumulation of NSRP, which perfectly co-localized with cis1. So green and red becomes yellow over here. Now, if you give recovery, what we see that this thing perfectly get exported to the cytosols. Right? So this is perfect that these things are usable, they are preserved, but they can be used. But now if I deplete cis1 with the help of this cis1 anchor array approach, they are accumulated, but they cannot be used up. They are not going to the cytosol at all. So this liquid to solid transition will not be available for the cell to be used up. So if this cannot be used up at all, then the cell will not grow properly, right? The cell growth will be perturbed for sure. So coming back to the growth report and cellular institution philosophy, that if we have normal, if we give cell heat shock and allow them to recover, they can perfectly recover. There's no problem in their growth because the chaperones are helping the nucleosynthesis ribosomal protein to keep it liquid. But if we deplete CIS1 and the NSRPs become solidified, there's a retardation in the growth, and it takes almost 500 minutes delay for them to reach the normal growth and to clear them up. So there is perfect growth advantage of this, keeping the stuffs in liquid form in that compartment. So what I'm telling to you is all about the story which we have found in yeast, right? What about human cells? Does this kind of things also happen in humans? So we look for a human parallel of CIS1 called DNAJB6. NPM1 is a nucleolar marker. So we can see the tiny dot nucleolus. We're looking at a super resolution microscopy over here, and we can see nucleolus forming a ring-like structure. Human cells perfectly grow at 37 degrees centigrade, so it's non-stress condition for them. And you can see that in the zoom image that this is the nucleolus, and DNAJB6, the parallel of CIS1 is everywhere, right? Except at this boundary, except at this perinuclear region. Now, if we give heat shock, which is about 42 degrees centigrade, 5 degrees above the normal condition, but we see that this particular vacant space is now filled by the CIS1 condensates. Now, as I told you that in yeast, we found that it's all about nucleosynthesis ribosomal proteins. So there's a drug called taurine-1. Addition of taurine-1 acutely deplete the production of any ribosomal protein in the system. So if we do the taurine-1 treatment, but we see that there's no further high condensation of CIS1 at this perinuclear structure. So if we draw a line over here, we can see that there is no overlapping of DNAJB6 with the nucleolus in non-stress condition. During heat shock, there is a perfect overlapping. But if you treat the cell with the drug taurine-1, even though we are giving stress to the cell, there's no accumulation at all. So we are basically saying that this whole mechanism is conserved from yeast to human, and it's not about temperature sensing, it's about sensing the process itself and accumulation of NSRP. So now with this, I can draw a model of sticking together to survive a stress model, where we have a system in a non-stress condition, CIS1, a HSP40, keep 70, repress all the time. When there is, you can see over here, CIS1 in the green, it's in the nucleoplasm, away from nucleus. When we give heat shock, CIS1 moves from nucleoplasm to this perinuclear structure. When it moves over here, this repressive effect is away and HSP1 can be activated. And this localization, I showed you, is sufficient, right? We don't need any temperature, only this localization is sufficient. And I showed you also that here, nucleosynthesis ribosomal proteins accumulates, and CIS1 try to keep it preserved over there so that during recovery, they can be used up. So this is what the NSRP looks like with the CIS1 being accumulated. And you can see the yellowing of NSRP with the CIS1. And if the cell have less ribosomal proteins, there's no polarization of CIS1, and cell, it doesn't matter if even though we are giving heat shock to the system. And all this processes which we found in yeast is conserved in human cells also. We give heat shock to human cells, and DNAJB6 also get accumulated at the perinuclear. But if the cell's producing less ribosome, it does not. So sticking together at this subnuclear region is very much vital for the growth of the cells, either it's yeast or human cell, so that during the recovery, they can be used up, not being discarded or not being degraded up. So with this, I'm going to thank my PI, David Pincus, for allowing me and giving me the opportunity to work this kind of a study, and also my lab members, also Anup sir and others for giving me opportunity for really talking and presenting my work at this platform. With this, I'm open for any questions, and thanks a lot for this opportunity.
Thank you, Dr. Asif Ali. Really, the presentation and the talk that you have given is very enlightening, is very interesting. Many things which were not known to me, especially I can talk about me. I've learned many things, especially the heat shock and other stress that you were talking about, that the newly synthesized ribosomal protein, how they condense at the nucleolar periphery, and then how they are activating the HSR. It's really interesting. And then you were talking about the movement of the HSP17 from the nucleus, and that is within 2 minutes. So that's really interesting, that how they are organizing within such a short span of time, and then they are taking control of everything.
Yes.
So, and then the very important thing is how the phase holds an important place, that liquid nature has to be maintained, and whenever you have the depletion of that phase, then the proteins are not acting. So basically you are losing the survival strategy.
Yes.
So
we're talking physics in the biology.
Yes, exactly. To maintain the state. So if you have a liquid state, you maintain it, you have a survival strategy. And if you lose that state, you miss the survival strategy.
Yes.
Very, very nicely presented by Asif. Thank you, Asif. So I think, let me see the chat box. I think we have, okay. Shudeshna ma'am wants to talk to you. Shudeshna, over to you.
Thank you, Madhumita-di. So, Asif, I can only tell you, yes, the talk was wonderful. And still I can remember when you were presenting the critical review presentation or the dissertation. So far, I can still remember you wrote so many chemical reactions and chemical structures. Okay. And during your MSc periods, you also gave very nice presentation, and the core subject was chemistry. Now you were joining physics with biology, and then on that time you had actually correlated the hardcore chemistry with biology. I can still remember those days. So best wishes.
Thank you for remembering me, ma'am.
Really, we are really proud to see Asif in this platform. Thank you, Asif.
This is really a great opportunity.
So any questions from anyone? Do we have any questions? If any of the students, you have any questions you want to ask, you can put it down in the chat box.
Asif, I have something to say.
Yes, sir.
It is good. You presented a fantastic paper on biophysics, but I really want that some words of wisdom and advice for our students. Because at the end of the day, I still find that people, after completion of their undergrad, they opt for institutes and they opt for other universities, considering that St. Xavier's is not a fit place to study, to pursue our postgraduate education. But unlikely, you as our first batch student, you actually stayed with St. Xavier's, you came to St. Xavier's by choice, and you pursued and you have reached certain height in your career. So some word of wisdom and advice and maybe guidance from you, which I actually really want for those who are attending both in YouTube and also in Zoom. Okay.
Yes, sir. It's really being, I can say that my whole career got changed when I joined St. Xavier's College, because initially the way I was looking biology is on white page, black letters. But now in St. Xavier's, I understand biology at practical levels, and the way that things are being teased is not only you are going to memorize the stuffs and this. We just got the examples that I remember Riddhi Majumdar sirs and Seshnam and others, and even Anup sir, they just teach us, I can say that teach us the biology as a mathematical problem, something, and to solve it really. It's not about, and it's not about memory, it's something beyond. And it's not only about that, it's the environment itself is so much student friendly for me at that time. I got selected in Rashtraguru Sundarnath College, where I think Madhumita ma'am was HOD at that time. And then
yes, exactly. I remember. I remember.
Yeah. Then she moved over here. Then I thought, okay, what's the reason going there if Madhumita ma'am is not there? So, but then I thought, okay, that's the really kind of a very, very great platform. And then I went, so for pursuing PhD, you have to go for this NET examinations, and I sat on the exam just 1 week after the MSc last semester exam of mine. So basically I have no preparation at all, zero preparation for NET, no tuition, no coaching. I just sat on the exam, what I learned in the 2 years. And that is my first click, and I got the CSIR qualified, right? With a rank of, I think I got rank 25 or 26. So basically, if you just follow what the curriculum of MSc St. Xavier's you have, you can just easily qualify that one of the challenging exams, which most of the higher. So what I did at that time is that I just stick. What I talk to you is stick together with all my current faculties and stick together with St. Xavier's alumni, and then they are really very, very helpful. And the first exposure I got, because it's not only about the way they teach us the theory in a practical, but also the way the practical itself being conducted in St. Xavier's is totally different. And it really make me to find much more interest to perform real experiments. So this 6-month exposure to Bose Institute in Ridhiman Sir lab was such a great, and that time I got the kind of interest that I should go for research, right? And then I should find something new. And throughout, always Anup sir, I mean, if I call him, he remembers me. So, and also as Deshna ma'am is saying that she remembers those talks. So it really is, I must say the environment matters, and the healthy environment St. Xavier's gives is, and just sticking to the curriculum they have, you can easily qualify NET. You don't have to really go anywhere else. And it's really, really such a amazing platform. And most of the things at the basic level I learned is from the St. Xavier's College itself. So it was really totally a game changing for me. And I really, I was one of the last students to be, or one of the, I was not in the first merit list. And, but I got selected because some of the students left, but I can say proudly that I was one of the first 2 students who got CSIR, and first student from that batch to got PhD degree, and then first to get the postdoc. So you can understand, my example can be a good example because I am not very well high academic before, right? Because I was not in the first list. But the way St. Xavier's trained me throughout, that now I am the first, not only first in that batch, but first among the first 20 in the whole country, basically, to qualify the CSR. So, and for sure, no preparation, just sat on the exam as it is, and then I was qualified. So, and I'm feeling very much proud to be the part of the St. Xavier, and it's really growing very well. I can see it's really growing very, very well, and then it's gonna make it much more better and produce much more better thing in future for sure. And thanks a lot for all those helps and supports.
Thank you.
And let me add up here. Asif was awarded Professor B.B. Bishash Outstanding PhD Student. As far as I remember, that was way back in 2017, isn't it, Asif?
Yeah, man.
Yeah. So in Bose Institute, that was 101 Foundation Day of the Bose Institute, and he was awarded that for the scholar. So good going for the Asif.
Yeah.
All the best.
Thank you, ma'am. I can see in the chat, Ridhiman sir asked about.
Yes, Riddhi sir is asking you, are you doing single particle tracking?
Yes, sir. Sure, we are doing single molecule particle analysis. The movie I showed and the analysis I have, that's the single molecule. The condensate moving, each pixel analysis, the analysis is done for the single particle analysis. And that's as the question is, that's very important to confirm that they are really intermixing and they are dynamic.
Okay. So any other question from anyone? Okay, I don't see any question. So Asif, thank you. Thank you very much. Hope to see you soon, very soon. We want to see you in the department. We want to see our Asif in the department. Thank you, Asif.
Thank you.
Thank you.
Thanks, ma'am.
I would like to, yes, ma'am. I would like to request ma'am to kindly felicitate Dr. Asif Ali.
Asif, this is for you. It is our honor to have you, Dr. Asif Ali, as a guest speaker in the Frontiers in Biological Sciences that is organized by the Department of Microbiology on 25th of March, 2022.
Thank you, ma'am. Thanks a lot.
Thank
I think
you.
I'm the first from that batch also.
Yes, exactly. So first in this also.
Selected last, but doing first.
But first, that's the tortoise and the rabbit race, Asif, isn't it? You have to be first. Whatever you have been, let's forget everything. But now you are in the top of the academic career.
Thank
All
you,
the best, Asif. All the best.
Thanks a lot.
Okay. So over to Srinjika for the
Thank you, ma'am.
program.
Thank you, ma'am. I would like to thank Dr. Asif Ali for delivering such a thought-provoking presentation to our audience, and our moderator, Dr. Madhumita Moitra ma'am, for wonderfully conducting our third and last technical session for the day. We have now come to the end of all the technical sessions for Fibs Chapter 5. Request my co-MC, Ananya D, to take over. Over to you, Ananya Di.
And with that, we have come to the end of the third technical session of Frontiers in Biological Sciences, Chapter 5. And now I would like to request Dr. Arup-Kumar Mitro, the convener of FIBS, to please give a formal vote of thanks and end the seminar. Over to you, Akin sir.
Thank you very much. Thank you for being with us, all the speakers, namely Shaurodipto, Sreyoshri, and of course Asif. Really, when I listen to your lecture, I feel that yes, I am getting old. But at the same time, I gain a lot of wisdom through your lectures, deep insight about biological sciences. Yes, you are right, Asif. The environment has not changed. It is absolutely the same, a closely knit family. Only maybe I am not there in the department right now because of my administrative position. But at the same time, in every way, I try to be with the department. And it is in my marrow that I always dream microbiology, Xavier's, that I call it in my blood, it is SXC, MCB, and it will be till the last breath of my life. I'm really grateful to you, all the guest speakers. Once you were student, but yes, you are showing us the pathway, the future pathway, the research is going on now. And I really feel yes, we are in the right track, producing a good band of students who will rule the earth, rule the biological science field in future. And definitely it will be like that, the two lines we all say, do you see what I see SXC, MC. And it is a fact that anywhere and everywhere in the world, I find this. And from the last 2 years, when we had this lockdown, obviously we were forced to webinars, but in a way, it has done good because we have stopped contacting external speakers when we have enormous talent within ourselves. So really, this particular Frontiers in Biological Sciences, Chapter 5 has proved it. I'm sure that all of us will admit that yes, SXCMCB has produced scientists and they will be paving the future world. I'm really thankful to Principal Reverend Dr. Dominic Xavier, SJ, because of his support. Because this particular program was organized in even less than 1 week's time. And equally, I am grateful to our Vice Principal, Professor Bertram Da Silva, for his support. And at the same time, the Dean of Science, Dr. Tapati Dotto. But above everything, I am really excited when I shared this with our head, Dr. Sudeshna Shamchaudhuri. She was the one who actually accepted it immediately and was proactive along with the professors there. And you can see the result. St. Xavier's Microbiology can organize a seminar of this kind in about 4 to 5 days. And it is only possible because of our able students. This is a legacy. I could find every time, whenever I have approached, I have seen the students rising to the occasion. I always say that each time, say after 3 years or after 5 years, students leave, but again they come back in a different name, and I am so close to them. So my request, they accepted it. And I'm really thankful to a band of students who made this possible. And that included definitely Shubham Jyoti and Boibhovi, the 2 student conveners. And we really, actually we communicated at odd hours of night just to ensure that things are moving in the proper direction. And really that shows the commitment. I'm thankful to you, Shubham Jyoti and Boibhovi. How can I forget your word? Sir, ar kichu korte hobe na to? This is the attitude. Yes, I like it. Spandon, your poster designing was tremendous. And it is because of that only we could actually spread across this message of Fibs chapter 5 to one and all. So thank you very much, Spandon. And Ritushri, with her able hand, she managed the registration and PR. And of course, Ahuna did the streaming, which is so important. But at the end of the day, a program only stands with proper comparison of the program. And this was done ably by Ananya and Srinjika. So I'm really thankful to you, dear students. I'm really thankful to all my colleagues in the Department of Microbiology. I'm thankful to the support staff. And yes, we plan, we think, and we execute. And this is the legacy of St. Xavier's Microbiology. And I believe it will continue in the future years. Thank you very much. God bless you. I think head should conclude the program.
Yes, sir. Thank you, Ikim sir, for giving the formal vote of thanks and thanking our students. Without the support of our teachers, Dr. Arup Kumar Mitro, our HOD madam, and all the teachers of our department, we could not have organized such a seminar at an international level without the support of yours. And now I would like to request our HOD madam, Dr. Shudeshna Sham Chaudhary, to finally take over and end the seminar. Thank you, ma'am.
So good evening, everyone. So it is time to announce the closure of this webinar. I want to thank all the organizers, Dr. Arup Kumar Mitro, all of my faculty members who have joined, who had moderated the sessions, as well as the well-known speakers, all of my student participants. Just I want to share my gratitude being a part of this webinar. Thank you all. Good night.
Thank you, ma'am. And with that, we would like to end the Frontiers in Biological Sciences, Chapter 5, 2022. And please like, share, and subscribe to our YouTube channel so that for any of the seminars, annual seminars we have, be it Modern Trends in Microbiology or Frontiers in Biological Sciences, you get updates and you can, attend, not miss out any seminars. And we have also given the link to the feedback form in the YouTube channel. So please fill it out, following which you will be receiving your certificates in due course of time. Thank you. And with that, we would like to end for today. Good evening and have a great day.